Om Shri Sai Ram, offering our collective pranams at the lotus feet of our most beloved Bhagwan, respected teachers and dear students. So I take this opportunity to welcome you all for the third session of the International Symposium on Sustainable Health, jointly organized by all the three campuses of Sri Sathya Sai Institute of Higher Learning, Department of Biosciences. In this session, we have to start with Sri Sanvit Pradhan, doctoral research scholar, working under the supervision of Dr. S. Venkatesh at the Prashantinilayam campus of the department, and he would be presenting his work on integrated omic analysis using data mining, conjures druggable pathways associated with Huntington's disease progression. I request Sanvit Pradhan to take over the podium and deliver his talk.
Offering myself at Bhagwan's lotus feet. Good afternoon and Sai Ram to all. So today I'll be speaking on how integrated omic analysis using data mining helps us to conjure druggable pathways which is associated with Huntington disease progression. So going on to the introduction, I would like to give a brief summary on what exactly happens in Huntington's disease. So Huntington's disease is a disease of the central nervous system and the brain. So in this disease, what happens is the Huntington gene is mutated, in which the Huntington gene comprises of CAG repeats, which over time or due to the mutation, there is an expansion of the CAG repeat, which ultimately leads to the misfolding of the protein and then leads to aggregation of this protein in the neurons, which ultimately leads to neurodegeneration. In normal cases, or the normal HTT protein or the Huntington protein genotype, we see expansion repeat of 10 to 35, which is normal. But in case of the disease, we see this expansion exceeding 35, which ultimately leads to the Huntington phenotype. As this is autosomal dominant disease, the penetrance of the trait is 50-50% amongst generation, and irrespective of the gender, it is passed on over. Also, I would like to highlight one more key thing, that is the length of the CAG repeat has a direct correlation with the age of onset. By age of onset, I mean the first point at which the phenotype or the disease progression starts, that is the cognitive function or the ability of the patient for motor functions deteriorates severely. And in normal cases, we see this around the age of 30 to 50 in case of Huntington patients. Also, I would like to highlight in the initial form of disease, the disease first strikes the striatum region of the brain, which is the portion of the brain which governs our motor and cognitive functions. Coming on to the symptoms of the disease has symptoms which can be classified into three, that is the motor, cognitive, and psychiatric symptoms. Also, apart from the neurodegeneration that is seen in the brain, Huntington is also known to be associated with many perturbations in many of the metabolic pathways, or also is related to oxidative stress and mitochondrial dysfunction. As we see in the slide, we see that HD is associated with lipid biosynthesis, as well as kynurenine pathway deregulation, vitamin B12, B6, folate deficiencies, inflammation, and hypohomocystinemia as well. So, from the point of Huntington being discovered in 1993, it has been more than 2 decades, and a lot of research has been done with respect to Huntington disease. But if we see the research that is being done, most of the focus is primarily on one aspect of the disease, that is either being transcriptomic, proteomic, or metabolomic. So what we have done is we have taken a systemic approach where we take into consideration all the omic techniques and then find a common deregulation that we want to explore. For doing so, what we have done is we do a data mining from various databases for each of the omic databases, that is GEO for transcriptomics, PRIDE for proteomics, MWB for metabolomics, and also literature search as well. So after we do data mining, we then do a differential analysis in which we employ various techniques like a GO2R function or network analyst, as well as metabo analyst, which gives us an idea about what are the differential expressed genes or proteins or metabolites in each of the case of the disease. Then with further processing of this data, we then go into selection of what are the significant pathways based on the p-value as well as the expression or the FDR. So further, we take these significant genes or the proteins or metabolites and then do a pathway analysis using various enrichment tools like Enricher, GSEA, and Klugo, as well as metabo analyst for metabolism data sets. This then gives us an idea what is the biological function of each of these differentiated genes as they cluster the genes into biological pathways. So these biological pathways are then imported into a pathway construction module where we use Cytoscape to visualize what are the pathways that have been deregulated, and with the help of various Cytoscape pathways or the applications that are plugged into Cytoscape, we do a clustering analysis and then find that what is the common element between all of this. We find what is the common element between all of these pathways. So once we know what pathways are being deregulated, using the betweenness or the commonness function which Cytoscape does, we find the key regulators for each of the pathways. This further is taken into model systems, like in our case in our lab, we have a model system of yeast and PC12, which we import the HTT, that is the Huntington protein or the gene inside the yeast using transformation techniques, and inside we transfect the PC12 as well. Then various knockout studies are carried out for each of these model systems. In case of yeast, also various nutrigenomics supplementation experiments using various vitamins as well as cofactors are done to see what is the effect of the particular deregulated gene or deregulated gene in the pathway, and overall what is the impact on the phenotype of the disease, that is Huntington. As you can see in this slide where I've highlighted in model systems, I have shown here that we have got 3 constructs of yeast, that is 25Q, 46Q, 72Q, and 1 or 3Q. So these are nothing but the expansion repeats of the Huntington in the Huntington protein of CAG, and in case of 25 and 46 is considered as a control, and 72 and 1 or 3 as the disease form in case of Huntington disease. So what we do is, with the help of knockouts and all, we try to see whether this aggregation is being attenuated in the experiments that we carry out. So some of the favorable phenotypes that we look for is C and D, which is in which we see that there is a complete clearance of the amyloids, whereas in case of A and B, we see that there is no change with respect to any experimentation that we have done, or B, which leads to more aggregation of the proteins in yeast. So as we know that there is no cure for Huntington, but however, the age of onset and the elevation of the symptoms for the disease highlights as a key research topic. And to address this, what we have done is we have taken two data sets with respect to mice and humans, and then we have studied time-based study where we compare, in case of mice, 12-week-old mice and 28-week-old mice, where we see that 12-week is a non-symptomatic Huntington model for mice, and 28 being symptomatic. Similarly, in case of humans, we have taken two data sets, that is PBMCs from the pre-symptomatic patients and the PBMCs from symptomatic patients. So these both are data sets that we compare, and we find that there is a significant amount of differential pathways that we see at the end of each time period, where we see a lot of differential pathways being present in the later phase of the disease. Similar experiments has been done in case of yeast, where we culture the normal form, or to say the probable form of Huntington yeast, that is 46Q. We culture this we culture the yeast 46Q construct over a time period, where we see in the log phase that there is no aggregation, which confers a control phenotype. But however, when we keep this for a long time, or when we culture this for a long time, that is till the stationary phase, we see that there is an aggregation which is formed, which indeed proves that Huntington has got its association with the age as well. So what we are trying to tell here is the age plays an important role in the change of various metabolic profiles, which ultimately leads to disease progression. So in the terms of sustainable healthcare, we would like to highlight some of our research, some of our experiments which we have done with respect to various medicinal plants from various plants from Indian Ayurvedic. I would like to highlight that we have done various experiments with respect to various medicinal plants that are amla, ginseng, guggul, and what we see that when we take the crude extract of this and we test it upon the aggregation that is seen in Huntington's, we see that there is actually the attenuation of. We see that there is actually the attenuation of each of this attenuation of the foci, which is observed in case of the disease form, that is 72Q and 103Q. However, there has been highlighted as a role of medicinal plants in many neurodegenerative diseases as well as cardiovascular diseases. This is with respect to it being rich in flavonoids and polyphenols. So, as we know that Huntington disease has got one of its perturbations being mitochondrial dysfunction and oxidative stress, we see that flavonoids indeed play a major role in scavenging the free radicals as well as chelating metal ions, hence reducing the oxidative stress. Also, there have been research with respect to resveratrol and curcumin as some of the key flavonoids which have been identified and are being used as therapy in many of the neurodegenerative diseases like Alzheimer's and Parkinson's disease. So next, this slide, actually this figure just sums up my whole presentation, where we have done multi-omic analysis and taken the key regulators, then we bind it into different degenerated pathways. We find the key regulators for these degenerative pathways, and with the help of nutrigenomics and knockout studies, we then see what is the impact of these key regulators onto the disease and ultimately the progression of the disease as well. So this is what leads us to the experimental validation and ultimately leads to clinical validation. So yeah, this is the reference that I've taken for making the slide, as well as, and I also take this opportunity to thank first and foremost our revered founder chancellor Bhagwan Sri Sathya Sai Baba, my supervisor Dr. S. Venkatesh, Department of Biosciences, our Central Research Instrumentation Facility, for helping all of us indeed for giving all the equipments that we require for our experiments. Also, I would like to thank my ever supporting lab members for the constant support and encouragement, and also for funding agencies for making our research happen. Thank you, Sai Ram.
Thank you, Mr. Samvit, for your wonderful presentation on Huntington's disease and the kind of work that you're carrying out. Now we will throw open the house for any questions. Yes, we have a question here.
Saram Sanvit, brother.
Saram.
Excellent presentation. I would like to know about what are the techniques of transformation that are used with regards to yeast and mammalian cell culture in order to introduce the mutated Huntington gene into the organism.
Okay. In case of yeast, like transformation in our lab, we usually do through two techniques. That is the chemical method and the electroporation method. So that's how we do it.
For mammalian cells?
For mammalian cells, actually, we have standardized a lot of techniques, of which we have actually like practicing too. That is the use of lipofectamine that we use for transfection of PC12 cells, as well as electroporation we do.
Is the chemical aspect of transformation, does it use the regular calcium chloride method or any other specialized method, especially for yeast?
Yeah, it does use the normal calcium chloride.
Or lithium?
Yeah, that is also there. Also, we also take the help of heavy DNAs, like from the sperm DNA from fish. So salmon sperm DNA. Salmon. So that is also there.
Oh, okay. For effective delivery
of
the mutant
correct,
gene.
Correct.
Thank you.
Thank you so much.
Sairam.
Yes, sir.
I have one question.
Please go ahead, sir.
So the question is, what are some of the findings with respect to the Huntington's disease?
So actually, so we are in the verge of like publishing all this data. So actually, when we do this transcriptomics, metabolomics, and proteomics analysis, integrated analysis, we do find some of the genes with respect to the TCA pathway, as well as many in terms of the glycolysis and all, to be deregulated as well, sir.
And also, I have another question.
Yeah, sure.
Why did yeast? Why was yeast moved for the studying the Huntington?
Oh,
Why
yeah.
not any other prokaryotic organism?
Yeah, first of all, like in case of prokaryotic organisms, since it is it doesn't have nucleus and all. So, and since this disease is to do with the higher organisms with respect to eukaryotes, so actually you have posed a very important question in this regards of why yeast is used as as a model system for neurodegeneration, as yeast doesn't have a brain itself to tell. So, apart from the cognitive functions or the behavioral studies that can be done with higher organisms like mammals or even monkeys or sheep, yeast, first of all, is a very cheap model organism, and it can be manipulated very easily, as the whole genome is known, and also we do have a full gene knockout of yeast with us. And what is the main point here of why yeast is used as a model system is all the major pathways to tell, like glycolysis or fatty acid degradation. So all the major pathways which are involved in many of the systemic diseases are well conserved in yeast and are as same as that seen in higher organisms as well. So there you have it.
So there is one more question that is posted on the chat box. Why yeast Q445? Why yeast Q46 considered as control? Because more than 35 Q repeats, we are considering it as disease in humans.
Yes, yeah. Actually, you've pointed out a very good question. So, actually, to tell that 46 is a kind of not a control, it is a disease form. As I earlier mentioned, that 36, whatever is there, which is beyond 36, poses disease in case of in case of humans. So, 46. Readily to tell, like readily 46 doesn't confer the phenotype. To tell, the readily 46 doesn't cause a ready aggregation. So when we do a time-based study, in case of yeast, why we take 46 is we want to see the aggregation in a period of time, however, which is not possible in 72Q and 103Q, as they form the aggregate very quickly. So a time-based study cannot be done with 72Q and 103Q, and hence we employ 46Q for our experiments with respect to time period. Yeah.
Thank you.
I have a question, sir. One question.
Please go ahead.
If the pathway which were, for Huntington disease in each model, the same pathway might be there, the same pathway in disease, that will not interfere with that. How that will relate to it? How is it different from that?
Sorry, ma'am, I couldn't follow.
The question is, you're talking about 46Q and 72Q. So you have a yeast model having this thing. But as such, yeast also prepares its own glutamate. So that will not interfere anywhere in this particular process when you're studying Huntington in the yeast model.
If I may clarify, ma'am is trying to ask, like you are trying to have 46Q repeats, etc. And the yeast might itself have, like it needs to produce glutamine, and then there will be a pathways where glutamine is incorporated into the proteins. Don't you think some of the housekeeping functions of yeast interfere with the kind of clone that you're transformed into the yeast? Will that affect your studies? So,
actually, whatever construct that we are using, it is it has been well used in many of the experiments with respect to yeast and neurodegeneration. For as it is extra chromosomal DNA that we are infecting the yeast with, I don't think so that any. And since it has got a promoter and promoter for itself in the plasmid, I don't think so it has any effect. Till now, there hasn't been any report with respect to the 46Q having any effect with respect to the glutamine utilization in the yeast itself. So actually, that is very a good question. I would like to. I'll get back to ma'am when I'll just look into it, and I'll get back to ma'am on this. Yes, ma'am. Yes, ma'am. Thank you, ma'am.
Thank you, Samvid. One last question from here.
Yes.
And you were mentioning about medicinal plants.
Yes, sir.
And more so, you pointed out towards flavonoids.
Correct.
So, what do you think would be the effect of flavonoids on, like, basically disintegration of the amyloids? Do you think is there?
Yeah, since like flavonoids are, first of all, it is an strong antioxidant, and it has its function in iron chelation as well. So, one of the deregulation that we see in Huntington is oxidative stress. So, flavonoids being a strong antioxidant, it has the capability of first of all reducing the oxidative stress on the mitochondria. And mitochondria, throughout the literature of Huntington, has been highlighted as a major key regulator of the progression of the disease, apart from the neurodegeneration. So I think so. Yeah. So I think so that actually we are just working on how flavonoids has an impact on gene interactions, and how it ultimately leads to disturbance in many of the metabolic pathways. But to what I know till now through my research is that flavonoids have a antioxidant property which helps in reducing the mitochondrial stress. So, which leads to elongating the age of onset for Huntington's disease.
So, if that is the case, have you considered screening with a library of flavonoids?
Yes, yes, brother. Actually, one of my paper I'm working on it, which gives this explanation, where I've been working with the protein interactions, as well as the genetic interactions with the flavonoids which interact to each of the proteins. Yeah.
Because I'm aware of a company called Select Chem.
Okay.
So where they can actually supply us 96 well plates coated with different flavonoids at different concentrations. So where generally it's very difficult to get a wide variety of flavonoids in one place and then try to screen them. So where maybe you can future probably consider trying to test with that. So where you have a library of close to 1,200 flavonoids in one place.
Yes, sure.
And screen them. So something might come out of that.
Mm-hmm. Sure, sure.
Thank you.
Thank you. Thank you so much.
Presentation. Sairam. Moving on, I request Miss Isha Sai Madam from Anantapur campus to introduce the next speaker and continue the proceedings. Over to the Anantapur campus. Sairam.
Offering my humble pranams at the lotus feet. It's a great pleasure to introduce our alumnus, Dr. Swati Saha, who completed her BSc in BZC from Sri Sathya Sai Institute of Higher Learning, Anantapur campus in 1989. She then moved on to the Sri Venkateswara University at Tirupati for her master's in biochemistry. Following this, she acquired her PhD degree in biochemistry from the Indian Institute of Science, Bangalore. Thereafter, she trained as a postdoctoral fellow in the US for several years before returning to India to work at the University of Delhi South Campus. Dr. Swati has been at the Department of Microbiology at UDSC since 2005 and is currently professor and head of the department. Her laboratory studies molecular biology in two model systems, the protozoans parasite Leishmania donovani and the Archaea Picrophilus torridus. Their group has published their research in several peer journals of international repute. I request Madam to take over the stage. Sairam.
Good afternoon and Sairam, everybody. Am I audible?
Yes, ma'am.
Okay. So it gives me great pleasure to be here with you all today. And at the outset, I'd like to thank Pradeep and the other organizers for giving me the opportunity to be here with you all. And thank you, Dr. Isha, for introducing me so kindly. So, So I embarked on a career in biology research just about 3 decades ago when I began my PhD at the Indian Institute of Science. But however, my journey into science started much earlier, way back in 1984, when I joined Anantapur campus right after 10th standard as a student of the intermediate program. At that time, I had no particular goal in life. I only knew that I wanted to study in Swati.
We are unable to see your presentation, ma'am. If you have already
You
started.
don't? You can't see it?
No, ma'am.
But I have started sharing it.
Could you? Yeah. Now we are able to see the present. You can maximize.
Okay. Okay. Is it visible now? Is it visible now?
Yes.
Okay.
It's
Okay.
not
So-
in the presentation mode, madam.
It's not in the presentation mode?
No, ma'am. If
How
you can
is that?
click on the full screen mode.
It is in the full screen mode on my laptop. So let me try that. Now
Slideshow, madam. If you can start the slideshow.
it is in slideshow. Are you not seeing it as slideshow mode? No? Is it not in slideshow mode?
Yeah, it's maybe slight delay in the transmission.
Ah. What are you seeing on the screen now? Are you seeing Anantapur campus?
Ma'am, can you come out of the presentation mode, ma'am?
Come out of presentation mode.
Yes. In slideshow, there's a tab slideshow.
Yeah. I'm on that tab only.
No, ma'am.
Hmm.
On top those menu tabs, that presentation animation slideshow in PowerPoint presentation.
Slideshow over here, you're saying.
Ma'am, in that.
Play from start. Yeah.
No, ma'am.
Now, is it not better now?
Difficult to see.
What are you seeing now? Are you seeing the campus?
1 second, madam. We are just.
Should I?
Ma'am, in that slideshow, there is a setup slideshow tab, ma'am.
In the slideshow, is it?
Uh,
Yes, setup slideshow.
Yes, ma'am.
Okay. Yeah, it is selected, presented by a speaker full screen. At my end, everything is okay.
It should be presented by an individual window.
Browsed by an individual window. Okay, let's try that then. Now?
Sairam, if I may interrupt, Madam can please present in this entire screen mode and then go to the screen slideshow mode, then maybe we can see it. Sairam.
Yeah, so that's exactly what I've tried to do now. What is the situation now?
We were able to see only the half of the screen, madam. So it's not in full screen mode. So that is what.
How is that? I don't understand.
Sairam, madam, can you just come out of the presentation once? We'll quickly.
Can I just try to log in again in case it is?
Just stop presentation, madam. It is very simple.
Okay,
Now click on present now.
yeah. Come to a window, right?
Click on the window.
Yes, done.
Your entire screen.
Just give me a minute. I have to open this first. No, otherwise, okay, it is now open.
No, when you click on present now, click on entire screen.
No, but you see, that will
No, I'll tell you, madam. 1 minute. You come out of presentation once.
entire screen.
Yeah. And then you click on top share.
Mhm.
You select that and then click share.
Okay.
Now click on your PPT. Now you put a full screen start presentation.
Now I
Yes.
sorry about that.
I think
Every
because
time
we are used to Google Meet and you are used to Zoom, this confusion. Yes, ma'am. Sorry.
okay. So as I was saying, I joined Anandpur. You're seeing the campus now in full screen mode, am I right?
Yes, madam.
Okay, so it's all good. So I joined Anandpur campus in 1984 as an intermediate student. And when I first applied at the college, I actually applied for the commerce program because I really did not know what I wanted to do with my life. I only knew that I wanted to study in Swami's college. But when I landed at college, the principal ma'am, Dr. Hemlata, she called me and counseled me. And thereafter, I moved to the BIPC program. So really, I think she is largely responsible for the career path which I have traveled. I spent 5 years at Anandpur campus, right from my 11th standard all the way to my BSc program. And I think I can honestly say they were the most formative years of my life. I was 16 and I was there till I was around 21 years of age. And I think the three most important things that I learned while I was there was, first of all, the importance of the dignity of labor and the importance of hard work. Second, the importance of leading an honest and disciplined life. And the third was the importance of being kind and considerate to those who are less fortunate than ourselves. And I have tried my best to live up to these three things. With Swami's blessings and lots of hard work by our then teachers, I graduated from the BSc program in 1989 and thereafter pursued higher studies, first at SV University, where I did my master's in biochemistry, then at IISc, where I did my PhD, and then after several years of postdoc, have now joined the University of Delhi, South Campus's microbiology department. So my laboratory basically studies the DNA biology of Leishmania donovani. And today I'll be telling you a little bit about our studies with DNA replication in this parasite and what strategies we are now adopting in trying to look for new solutions to Leishmaniasis. So Leishmaniasis is actually a group of diseases which is caused by about 20 different Leishmania species. This is a digenetic protozoan parasite which shuttles between its insect host, the sandfly, and the vertebrate host. This parasite was first identified way back in 1903 by two British medical officers into independent studies. So the first was Sir William Leishman, who was in England and who identified these parasites as darkly stained bodies in tissue sections of spleen when he was performing an autopsy on a British soldier who had just returned from India and who had passed away. And he published his findings in the British Medical Journal under the heading of Trypanosomes in India. Now, later the same year, the other British medical officer, Charles Donovan, who was stationed in Madras, he reported that these very parasites were the causative agent of Kala-azar in Indian patients in Madras. And so thereafter now this new genus which was discovered was named after the discoverers as Leishmania, after Leishman, and Donovani, after Donovan. And the disease it causes came to be called Leishmaniasis. So according to WHO, more than 1 billion people across 88 countries live in areas that Leishmaniasis is endemic to, and they are thus at the risk of infection. About 12 million people are infected with this parasite globally, although many of them do not display any symptoms. In the old world, the disease is endemic to South Asia, East and North Africa, the Middle East, and Southern Europe, while in the New World, the disease is endemic to Central and South America. So Leishmaniasis are the second highest killer among parasitic diseases, the first, of course, being malaria, and is spread by the bite of the insect sandfly. This disease actually afflicts the world's poor who do not have access to clean sanitation, who live under very unhygienic conditions, and who also suffer from poor nutrition. So these three factors make them very vulnerable. So this disease is actually manifest in three forms: cutaneous, mucocutaneous, and visceral. And as you see, each form of the disease is caused by a different set of species of the parasite. So cutaneous leishmaniasis is characterized by erythematous nodules gradually ulcerating and resulting in severe scarring. The mucocutaneous parasites, these are typified by the invasion and destruction of the nasopharyngeal mucosa by the parasites. Visceral leishmaniasis, however, is the deadliest of the three forms of the disease, whereupon the liver, spleen, and bone marrow get infected. So there is hepatomegaly and splenomegaly, which is leading to this swollen abdomen that you see in the picture. And this is accompanied by severe anemia and weight loss. And if not treated in a timely manner, visceral leishmaniasis can actually be fatal. So in India, of these three forms of the disease, the prevalent form is visceral leishmaniasis, popularly called as Kala-azar, because of the irregular bouts of fever, which is accompanied by a darkening of the skin. According to WHO, about a decade ago, there would be almost 500,000 new cases of visceral leishmaniasis reported each year, and 90% of these cases were from the Indian subcontinent, Brazil, and Sudan. Today, however, this number has dropped considerably, and according to CDC, today there are less than 100,000 new cases being reported for VL each year, and about, of course, 1 million new cases of cutaneous leishmaniasis being reported. India is now close to, so in India, visceral leishmaniasis is actually found in the eastern parts of the country, and that is West Bengal, Bihar, Jharkhand, and eastern Uttar Pradesh. And we are close to eliminating visceral leishmaniasis thanks to our extensive surveillance programs, as well as several prophylactic measures which the government has put in place. But the new concern on the horizon now is the emerging problem of post-Kala-azar dermal leishmaniasis. So post-Kala-azar dermal leishmaniasis, or PKDL, is seen in patients who have recovered after visceral leishmaniasis after treatment, and this appears within 6 months to 2 years after recovery. This disease is characterized by maculopapular or nodular rash on the skin. Now, PKDL is not fatal, but however, patients in India will rarely recover from PKDL spontaneously, and thus these patients become reservoirs for the parasite. So it is important to control PKDL as well. So how do you diagnose the disease? There are several field tests available. The Montenegro test, which is a test where you inject a small amount of Leishmania antigen into the skin and then look for a delayed hypersensitivity reaction. The logic again being that, if you are infected, you have antibodies and that would interact and so on. There is a rapid agglutination test, dipstick test, where basically the antigen is immobilized on the dipstick. So again, you're testing for the presence of antibodies in the infected person. And then you have a large number of ELISA tests which are actually available, which are against various Leishmania antigens. And more lately, there are PCR-based tests as well. Now, these tests are very handy in the field because they are rapid. But the first 3 of these tests represents the problem of you continuing to get a false positive result even after the pathogen has cleared because of the persistence of the antibodies in your blood. The PCR-based tests are relatively newer of the lot, but even those are not always accurate. And therefore, even today, the gold standard test for Leishmaniasis is microscopic observation. So if it is cutaneous Leishmaniasis, you take tissue sections from skin lesions, or you draw out the aspirates from the skin lesions and make smears. And if it is visceral Leishmaniasis, then you take tissue from the liver, you do liver biopsy basically, or you make smears of the splenic and bone marrow aspirates and then analyze those for the parasite. Observation is of course done by standard staining. Either you do GIMSA staining where the parasites stain dark purple, or you can even do immunoperoxidase staining. Prophylaxis. As far as prophylaxis is concerned, to date, there are no vaccines to this disease, and this is not due to lack of trying. Several groups are on it, but so far, no success. There are still several candidates under preclinical trials, and there is one particular vaccine, Leishf3, which has now completed phase 1 clinical trials. So most efforts in prophylaxis actually relate to preventing the proliferation of the insect host and protecting us from the insect host. So this is again a lot like what you see with malaria, where there are no vaccines to date. And so when we say we want to protect ourselves from malaria, we basically see how to get rid of the insect vector host, right? So either we use bug repellent or mosquito nets and so on and so forth. So the concept is the same here too. The aim is to do away with the sandfly, and if there's no sandfly, there's no propagation of the parasite. For treatment, these are the currently available treatments. The first line of treatment is the intravenously administered sodium stibogluconate. This is a pentavalent antimonial compound, which is administered intravenously. However, because of emerging drug resistance now, for a long time, actually, this was the only treatment. But because of emerging drug resistance, now alternative treatments have come into the market. And in India, the current treatment is the oral drug, miltefosine. This drug was actually developed as a therapeutic right here in India at the BHU by Dr. Shyam Sundar's group. WHO's recommended treatment is the liposomal formulation of amphotericin B. Now, although these treatments are available, they are expensive. The treatment regime is quite long. These treatments all continue between 30 to 40 days. And moreover, they all have toxic side effects. Add to this the fact that there is constantly emerging drug resistance. So therefore, the search for new sites for therapeutic intervention continues, and several groups around the world are involved in studying the cell biology of this parasite in the hopes of identifying new drug targets. So this parasite actually is digenetic, living in the insect host as these flagellate promastigotes, which eventually migrate to the insect's proboscis and gets released into the bloodstream of the human with the insect bite, whereupon they are phagocytosed up by the macrophages, within which they now transform into these tinier, roundish mastigotes, which are flagellate. Actually, they do have flagella, but very tiny if you look at the cartoon on the left-hand bottom of the slide. So for practical purposes, we say they're flagellate because they're non-motile. So these mastigotes, they multiply within the macrophages, eventually get released from them, get taken up by the surrounding macrophages, and thus the cycle continues. So when the insect bites the infected host, it takes in these infected macrophages as part of its blood meal, and then in the insect's midgut, again, the mastigotes get converted to promastigotes, and the cycle continues. So both promastigotes and mastigotes actually reproduce asexually only by binary fission, and DNA replication is central to this process. DNA replication is broadly conserved between prokaryotes and eukaryotes in that it's a semi-conservative process where each strand serves as a template for the synthesis of the single daughter strand. Now, because prokaryotic genomes are circular in nature and they have a single chromosome, there's a single origin of replication called the ODC, which carries these consensus sequences called the A boxes. And the DNA A replication initiator protein binds to these sequences, melts the origin DNA. This melted DNA now gets coated with with SSB protein, so that the two strands do not come together again. And thereafter, the DNA B helicase gets loaded along with the other replication enzymatic machinery. So the helicase unwinds the DNA duplex as the fork advances, and the polymerases and other associated proteins are involved in the synthesis of the DNA strands. And as you see over here, along one strand, you have continuous synthesis, that's the leading strand, while on the other strand, you have discontinuous synthesis, that is, the strands are synthesized as short Okazaki fragments, which eventually get ligated together to give you the long single strand. When you look at DNA replication in eukaryotes, of course, every chromosome has multiple origins. But interestingly, other than Saccharomyces cerevisiae, none of the other eukaryotic genomes carry any consensus sequences in their origins of replication. So it's a mystery as to how the initiator protein ORC really recognizes these origin sequences. What is known now is that genome context plays a major role, and origins are typically AT-rich, but consensus sequences have been identified only in Saccharomyces cerevisiae. DNA replication in eukaryotes is a highly conserved process where the actual process of DNA synthesis, which occurs in S phase, is preceded by the licensing of origins in the G1 phase by the formation of pre-replication complexes either at or very near these origins of replication. So the pre-replication complex formation begins with the association of ORC, which is depicted by this very large green oval in this cartoon. So the ORC complex, which comprises of ORCs 1 to 6 proteins, associates with the origins, and then thereafter sequentially recruits CDC6, CDT1, and the MCM2 to 7 double hexamer. And the association of this double hexamer now completes the process of licensing, so that as the cells enter S phase, specific components of this MCM2 to 7 hexamer get phosphorylated, leading to the recruitment of Cdc45, GINs, and the DNA polymerase. And this is followed by the splitting of the double hexamer, so that two replisomes now move apart in opposite directions, synthesizing DNA as they move along in a template-dependent manner. So what about in Leishmania? Yeah, and thereafter actually the replication machinery is very similar between prokaryotes and eukaryotes. The actual enzymes and proteins which are involved may be different, but they are very orthologous and homologous in sequence as well as in structure with what is seen in prokaryotes. So what about in Leishmania? When the Leishmania whole genome sequence was published, it was found that again the sequence, the process is broadly conserved in Leishmania as well, with several proteins being identified like the ORC1, the MCM2 to 7, and Cdc45. However, several key components were also absent, like the ORCs2 to 6, and there was no distinct CDC6, nor has any distinct CDT1 been identified so far. Furthermore, the GINs and the replicated polymerases are also somewhat different in their structure. So the absence of several of the normally conserved components of these pre-RC and replication process suggests that novel Leishmania-specific proteins may actually be involved in the process. And so the long-term goal of our laboratory's research is to use the Leishmania orthologs of known eukaryotic replication proteins as bait in proteomics analysis to fish out the novel proteins that may be involved in the process. And such proteins would then be tested for being putative sites for therapeutic intervention. So the focus of the study I'm presenting today is the Cdc45 protein, which is a part of the CMG complex. This is the replicative helicase complex, whose core component or core helicase is the MCM2 to 5 heterohexamer, which is able to encircle the template DNA by opening up this little gate between two of the subunits, the MCM2 and MCM5. And once this heterohexamer is encircled the DNA, this gate needs to be not only shut, but it needs to be locked and barred in place so that the helicase can move along smoothly along the template ahead of the advancing replication fork, unwinding the DNA. And that is where the role of Cdc45 and GINS comes in. These are the two proteins which are responsible for locking the gate and keeping it shut. So since Cdc45 is a conserved protein, of course, we expect that in Leishmania as well, it would be involved in DNA replication. But we began our study with first checking if this was indeed so. And the approach we used was to deplete the cells of Cdc45 and then look at the replication phenotypes obtained. Now, because Leishmania doesn't have the siRNA machinery, we were forced to resort to making genomic knockouts, and this is standard in the Leishmania field. Another unusual thing about Leishmania is that it has a very plastic genome, and some chromosomes are actually trisomic. And Cdc45 actually lies on a trisomic chromosome. So what we have attempted to do is to knock out the 3 genomic alleles sequentially, one by one, where in the first step, one of the alleles is replaced by a drug resistance cassette, and the cell line so formed now becomes the recipient for the second step of homologous recombination, where the second allele is replaced by a second drug resistance cassette, and the cell line now formed becomes the recipient for the third step, where the third allele gets replaced by a third drug resistance cassette. So the way you do this is, of course, you create a donor plasmid where you put in the flank sequences of the gene, and then you linearize the plasmid and resect the cassette, basically the donor cassette, and you transfect it into the parasites, and then you plate out your transfection mixes, screen your colonies, and then you move on from there. So we found that we were able to successfully create replacements of the first genomic allele as well as of the second genomic allele, checking for pristine recombination at both ends by PCRs across the deletion junctions. But we were unable to create a true Cdc45 null. We were only able to replace the third allele in cells which were expressing the Cdc45 protein episomally from a plasmid. So what this is telling us is that this protein, therefore, is essential for cell survival. And as we were unable to create a true Cdc45 null, we then moved ahead with our phenotypic characterization using the heterozygous knockout cells. And so then the first order of the day was to see how the expression of Cdc45 was impacted by the knockouts of these single or double alleles. And what we found, when this is a real-time PCR experiment, where we found that when you lose one allele, expression goes down considerably, and that is the pink bar over here. But when you lose 2 alleles, expression goes down dramatically, as you see in the red bar here. So to study the impact of this Cdc45 depletion on the parasite, we first carried out a basic growth analysis study. And what we found was that growth was severely compromised when you knocked out either a single allele, indicated here by the pink line, but more so when you knocked out 2 alleles, indicated here by the orange line. And furthermore, when we looked at the generation time, the double allele knockouts had a much longer generation time. It was 16 plus hours as compared to 9 plus hours for the wild type. So now to see if the generation time, the lengthened or the increase in generation time was linked to the lengthening of any particular stage of the cell cycle, we now studied the knockout cells for their cell cycle profile using flow cytometry. So flow cytometry analysis will allow you to sort a heterogeneous population of cells one cell at a time on the basis of the characteristic fluorescence and light scattering of each cell. So how do you use this method to study the cell cycle? You basically stain your cells with propydium iodide. What propydium iodide will do is stain the DNA component of the cells. So how do you identify the cell cycle stage based on that? The logic here is that in G1 phase, your DNA has not yet replicated. By the time you go to G2 phase, the quantity of DNA in the cell has doubled because it has now replicated. So what do the flow cytometry profiles look like? If you look at these panels over here, the peak at the 200 position, these are the cells whose DNA have not yet replicated. So that is your G1 cells. If you look at the last panel, those are the cells where the DNA has completed replication. So now all the cells are at the 400 position because the DNA component has doubled. And the cells which are actively replicating, which are in S phase, they are now spread between the two peaks. So when you're looking at an asynchronous culture, what you're seeing is majority of the cells are in G1, that is your log culture. Some cells are in G2M and the rest are in S phase. So how did we analyze cell cycle progression in these knockouts using flow cytometry? We basically blocked the cells at the G1S transition over here by the addition of hydroxyurea, and then released the cells from the block and allowed them to navigate across S phase, G2M and G1 to follow how they were moving across the cell cycle, harvesting cells at different time points and then fixing them and then staining them with propydium iodide to monitor their cell cycle status. And so what we found was, so before your eyes glaze over, the very top row is the wild type. The middle row is basically the profile where you've just knocked out one allele, and the lowermost row is where you've knocked out two alleles. So if you just concentrate on the red box here, what you see in the wild type scenario is that once you've relieved the block, the cells are moving into S phase very smoothly across into G2M and back into G1. You knock out one allele coming one row below, the cells still enter S phase very smoothly, but then gradually slow down, taking slightly longer to come back into G1. But when you knock out both alleles, there is a very dramatic effect where now the cells have difficulty even entering S phase and generally take much longer to navigate S phase and come to G2M. So these data basically affirmed that the Leishmania Cdc45 is functionally conserved in that it plays a major role in DNA replication. Now, when we first cloned out this gene, we noticed that it carries this 7 amino acid motif called the PIP motif. The PIP motif is a motif which is typically found in most proteins that interact with PCNA, PIP, PCNA interacting protein. So what is PCNA? PCNA is actually the eukaryotic ortholog of the bacterial beta clamp protein. It is a protein which is responsible for clamping on the polymerases onto the template DNA so that DNA synthesis can occur efficiently. So PCNA is an essential protein. Without PCNA, replication cannot happen and the cells will not survive. PCNA is the clamp protein not just for the replicative polymerases, but also for the repair polymerases. And in addition to serving as the clamp protein, it also interacts with several other replication and repair proteins. This is a homotrimeric protein, and because each monomer carries this inter-domain connector loop, therefore, the trimer can simultaneously interact with 3 different partners if need be. The binding partners which interact with PCNA do so via their PIP box, which gets docked below this IDCL. Now, when we identify this PIP motif in Leishmania, what struck us was that there were no other reports to date about Cdc45 PCNA interactions. Although a large number of binding partners had already been identified for PCNA, Cdc45 was not one of them. So we immediately looked at other eukaryotic Cdc45s to see if they had the motif. And interestingly, we found that Saccharomyces cerevisiae Cdc45 does not carry this motif, but the human Cdc45 does. Now, the human Cdc45 crystal structure was published in 2016. So what we did was we mapped this PIP motif on that crystal structure and found that it lay in this alpha 12 helix. That's the little red helix in the midst of the blue. That's the PIP box. So all of the blue helices over here represent the face of the protein, which is involved in interactions with the MCM2 to 7 core helicase. And since PIP is a part of that interface in human Cdc45, it is unlikely to be involved in any other interactions. So what we did was we took the Leishmania Cdc45 sequence and now modeled it using the Fire2 tool against the human Cdc45 crystal structure and found that the structures were in fact very conserved, including the helices which are involved in interactions with MCM. But the PIP box now does not lie in the alpha 12 helix, but rather lies on the opposite face of the protein, suggesting that it may in fact be available for interactions with PCNA. So we then did two very simple experiments to test if indeed the two proteins were interacting. The first was an immunoprecipitation reaction. The concept of this experiment is very simple, as most of you must know. If you have an antibody to a particular protein, and that protein is actually in complex with several other proteins, then when you add the antibody to the cell lysate, it will not only bring down that particular protein, but it will also bring down all the other proteins that are associated with it. So this immunoprecipitate is collected and then analyzed by SDS page and Western blotting. And what we found was indeed, so what we did was we immunoprecipitated PCNA because we had antibodies to PCNA from an earlier study in the lab. And we found that the PCNA immunoprecipitates actually did carry co-immunoprecipitating Cdc45. So this told us basically that the two proteins do interact with each other stably in whole cell extracts. But then the question is, do they interact directly or indirectly? In other words, is it a question of the yellow circle interacting with the green bucket? Or is it a question of the yellow circle, oval, I beg your pardon, interacting directly with the red octagon? So to check this out, we did direct pull downs between the two recombinant proteins. So the way you do this is you purify the two proteins in the recombinant from E. coli, and now you incubate the two proteins together. One of the two proteins you tag so that it can be captured by an affinity ligand. So after you incubated the two proteins, you now add on the affinity ligand to pull down the tagged protein. And what you then do is run out the pull down, you elute the pulled down fraction and then run it out on SDS page and Kumasi stain it and see if you were able to pull down the interacting partner or not. So if you don't get a prey band, that means the interaction must have been indirect in the immunoprecipitation reaction. So when we did this direct pull down experiment, we actually found that the interaction was quite direct. So just concentrating on the last lane here. So when we pull down PCNA using the His tag and nickel affinity beads, the Cdc45 protein also got pulled down. Okay? And the rest of the lanes are all the appropriate controls which need to be included. So now it is clear that the two proteins interact stably in whole cell extracts and they interact directly. So the question is now, of course, remember the whole story started because we identified the PIP box. So is this interaction happening via the PIP box? So it was a simple matter of making PIP mutant proteins and then carrying out the immunoprecipitation and pull down experiments. And we found, so here just concentrating on the last two lanes, The second last lane shows the immunoprecipitation reaction where you are expressing the wild type protein in the cell. And the last lane shows the same reaction, but instead of the wild type protein, you're expressing the mutant protein in the cell. And so what you see instantly is that the interaction is lost. And this was also the same story when we did the direct pull down experiments. So the wild type protein once again gets pulled down, but not the mutant. So thus it is now clear that the two proteins are interacting directly via the PIP box. But so what if they're interacting? Is it even important, and does the cell even care? So to check this out now, we did a very simple genetic experiment where we took the heterozygous knockout cells, which had severe growth and cell cycle defects, and overexpressed the wild type Cdc45 and the Cdc45 PIP mutant proteins in those cells. So this is just a Western blot analysis, which is showing you that both the proteins were expressed appropriately. And now we looked to see whether the expression of these proteins could actually rescue the growth defects. And what we found was, so looking at the growth curve here now, the blue curve shows the heterozygous double allele knockout, which has compromised growth. When you express the wild type protein in these cells, now you rescue these growth defects, which is what you see with this pink or magenta colored line. But when you express the PIP mutant protein, you are unable to rescue these defects. And the same thing was examined in flow cytometry as well. And the first row represents the wild type, the second column, the knockout, the third column, the rescue with the wild type protein, the fourth column, the rescue with the mutant. And so what you can instantly see is that the first and third columns look similar. In other words, the expression of the wild type protein rescues the defects, whereas the second and fourth columns look similar. In other words, expression of the mutant protein is not able to rescue the defects. We also tried to actually knock out the third allele in a background which is expressing the PIP mutant, and were unable to do so. And so taken together, these experiments show that the Cdc45 PIP box is essential for cell survival, and the Cdc45 PCNA interaction is therefore critical for the cell. So why is it critical, and what is the role of this? We spent actually several months thereafter in experiments looking for answers to those questions. And I will not go into the details of the experiments, but will only tell you that at the end of it all, what we found was that Cdc45 seems to be actually helping tether PCNA to the template strand, thus facilitating DNA replication. So where do we go from here? We are now working towards identifying the novel binding partners of the Cdc45 protein. Again, how would we do this? We would use the same immunoprecipitation concept, where we would add the antibody to Cdc45. We have now tagged the Cdc45 protein, so we would immunoprecipitate it out and expect that along with it, all the other interacting partners would co-immunoprecipitate out. Except that now we are not searching for a known partner. So we are not doing a Western. Now we are looking for the unknowns. So what we would do is scale up the experiment, analyze it by STS page and silver staining, and then identify each of the bands which appear by mass spectrometry analysis. And once we've identified the interacting partners, we will then follow it up by making genomic knockouts to assess the phenotypes obtained, and any essential proteins which are thus identified will be tested further for being possible sites for therapeutic intervention. With this, I will bring my talk to a close, but not without acknowledging the funding agencies. DST and DBT have been my constant funding sources. I have occasionally had grants from CSR and UGC as well, But this particular grant, this particular work was funded by DST. These are all my students in this group picture. This work was done by my student Aarti Yadav. The structural part of the work was done in collaboration with Dr. Manisha Goyal, who is a faculty member of the biophysics department, because I don't have expertise in that. And the other people who worked on this project were my three students here, Varshini, Jyoti, and Pallavi. With this, I will stop and I will be happy to take any questions that you may have.
Thank you very much, madam.
You're welcome. Any questions?
Now we will open the house for any questions. Any question from the Anantapur campus, madam?
Sai Rao. Madam, I have a question. So identifying the interactive protein.
I'm sorry, I can't hear you very clearly. Can you speak up a bit? Hello, yes. Can you please speak a little louder?
Yes, ma'am.
Yeah, that's sure. Tell me.
Identifying the interacting protein.
Yes.
Are you not considering two hybrid system to see if you can identify?
So the two hybrid system would be done in a heterologous system. Okay, there is no Leishmania two hybrid system available to date. So we would either have to do it in Saccharomyces cerevisiae or we would have to do it in mammalian cells. So the moment we are doing that, now, you see, we run against the problem of these proteins being very conserved in those systems. Okay. Second of all, whatever we pick up in those systems will not necessarily be what is actually happening in Leishmania. That is why we are choosing to go via the route of immunoprecipitations, where we are completely doing the study in Leishmania.
Okay. Thank you.
Saira ma'am,
Yes.
excellent talk. Ma'am, I had a very naive question. So you said that you were unable to generate any null mutant where none of the copies are functional. So is it because of the lethality issue that without CDC?
Yes, that's in fact the very reason. And that's why I said that we could not make a true null. So if you don't get colonies when you make a genomic knockout, because how do you select?
Yes, ma'am.
Right. Now, if you don't get colonies on drug, that could very well be because you're a lousy experimentalist, or there could be a bigger issue. So the way you do this is you simultaneously put in your donor cassette into two types of cells. One is the double heterozygous knockout where you've already knocked out two alleles. The other is the double heterozygous knockout where you are already expressing the Cdc45 protein from a plasmid.
Yes, ma'am. That's what.
Yeah. So what we were able to do, so had both given us no colonies, that would have meant that it was an experimental issue.
Okay.
But however, we got plenty of colonies in the case where we were trying to make the null in a CDC45 plus background. And so therefore we conclude that the gene is essential and therefore we have not been able to knock it out.
And ma'am, is it not possible to generate any kind of conditional null mutant where under certain restrictive conditions and
yes. So in Leishmania, there are just about 2 or 3 reports about conditional nulls. And there's a lot of uncertainty about how well these experiments would actually work, because a couple of labs have got them to work, but in the vast majority, I hear that it's not that easy. I have to confess that our lab, we have not tried it. So I cannot say firsthand how easy or difficult it is. However, rather than, of course, a conditional knockout has an added advantage, but of late, what is also catching up a lot in Leishmania is making these CRISPR-Cas knockouts. So there are studies now coming out where people are actually doing CRISPR-Cas knockouts, and again under, you know, so again there the problem we would face is that if it's an essential gene, it may not survive. But now people are devising and working on ways to make all of these methods conditional. So I'm sure the field is moving very fast, and I'm sure in a few years, these procedures will also be very streamlined, and it should be reasonably easy to make those kind of lines as well.
Thank you, ma'am.
And posted on the YouTube chat box. Scientists have developed genetically modified mosquitoes to eradicate disease like dengue and malaria. So can we expect the same for sandfly in future?
So this is a good question and a loaded question. I don't know how I can answer that. So it's like this. Yes, we have made, I mean, scientists have made genetically modified mosquitoes. And if I'm remembering this correctly, a study in, I think it's in Florida, where they've actually released these in a controlled fashion to see if it would work. So far, there are no reports of this kind in sandfly, but there are several groups which are actually dedicated to studying the sandfly's biology and seeing how it can be controlled. So I think we just have to wait and see what happens. Of course, the moment we are making a genetically modified organism, you see, there are so many ethical issues which come up, and there's so much discussion on this and so many regulations on this. I'm not sure how far it would actually go, practically speaking. You know, so now, for example, you have the genetically modified mosquitoes, but how far could one go in replacing the wild type with these? So when it comes to that, then scientists are basically in the hands of the regulatory bodies. So, but however, I'm sure that that should be doable. That should be doable.
Thank you, madam. There is one more question that is posted, madam.
Yeah.
According to you,
Yes.
what approach should we use to design an effective vaccine against Leishmaniasis?
Yes, that's a very good question. I have to tell you that there are several groups who have been trying really hard to get a Leishmania vaccine going. And so there are lots of, you know, proteins which have been used or which have been tested as putative vaccine candidates at the preclinical stage. Okay? So, and usually these are actually the surface proteins because those are the ones which will be easily accessible. The most promising candidate, which actually also went through phase 1 trials, was this Leash F3. What they have done over there is actually they've made a recombinant antigen, which is a fusion of two different Leishmania proteins. And again, that was very promising in preclinical and was, you know, taken forward, in fact, by the IDRI. This is the Infectious Disease Research Institute at Seattle in Washington, which is actually dedicated to, you know, vaccine development for all these diseases, TB, malaria, these are their flagship programs. They also work with this Leishmania thing. So ideally, when you want to look for a vaccine candidate, you want to look at a protein which is on the surface. However, when you're looking for a drug candidate, then it's a different story, of course. Then it could be something which is cytosolic as well.
Thank you, madam, for the response. Any questions from the Brindavan campus?
Sarah, ma'am.
Sarah?
Ma'am, I wanted to ask that are there some new drug delivery targets and systems that are being worked upon with Kala-azar?
I'm sorry, your voice is echoing.
Sarah ma'am.
Come in again.
Sarah ma'am, am I audible?
Yes, now it's clear. Please tell me. Yeah. Come in, yes, please.
Sarah ma'am.
Yes, yes. Please go
Ma'am,
ahead.
what are the new drug delivery systems and targets which are being worked upon with respect to Kala-azar disease, since you have made a mention upon it?
Yes, with respect to Kala-azar disease, I will tell you right now. So in India, this was actually one of ICMR's flagship programs along with DNDi. There has been a major effort to eliminate visceral Leishmaniasis. Okay, and I think we are at a point now where last year I heard there were less than 10,000 cases reported. Of course, again, a lot of cases, WHO always tells us that a lot of cases do go unreported. So, however, now the focus has turned to PKDL, because what is happening is now they are becoming reservoirs. So, unless you get rid of the reservoir, and I don't mean the person, I mean, I beg your pardon, the parasite, unless you get rid of the parasites in the PKDL patients, then there is always a risk of this flaring up again. So PKDL right now is, there has not been too much work done on it. It is typically right now being treated by sodium stibogluconate and as well as with biltifosin. In India right now, there is a clinical trial going on, which is at phase 2 level. And what they're doing is they're trying to now look at a combination treatment of amphotericin B and miltifosin and seeing how that would work. So basically, whether it would be more effective, I think the idea is to cut back on the complexity of the treatment regime. So all of these treatments go on for 30 to 40 days. In the case of miltifosin, you're actually having to take 3 pills a day for those 28 to 40 days. In the case of sodium stibogluconate, it's an intravenous injection which you may have to take every day. With amphotericin B, there are regimes where you're going in for intravenous injections every alternate day. So in terms of a brand new drug, there are no actual published reports. Rather, what people are trying to do is repurpose existing drugs, because that would simplify the whole process of doing the trials and so on.
Sarah, ma'am.
Yes.
Am I audible, ma'am?
Yes, you are.
Ma'am, I want to know, as you told, this Leishmania is a digenic in nature,
Right.
genetic in nature. So how it, when it is phagocytosed by macrophage, how it is escaping the immune mechanism or the immune response of the macrophage? Ma'am?
Yes,
Is
that's
it clear or some same mechanism is there?
Yes, yes. This is a very good question. So, and that is why I said that the world's poor are the most vulnerable, because they have weak immune systems. So several of us, I shouldn't generalize, because in our country, Leishmania is only in certain pockets. But there are a large number of people who are actually infected. It's kind of like tuberculosis, right? You're a carrier until you show symptoms. Okay, so your immune system fights back. Now, how does Leishmania, in the cases where Leishmaniasis flares up and where our immune system fails us, the way it does it is it actually suppresses the immune response and somehow increases the production of IL-10. IL-10 is a cytokine with anti-inflammatory properties, which limits the immune response. So somehow Leishmania manages to trigger an enhanced IL-10 response. That is one way. Second of all, so again, if you look at what's happening in the macrophage, so Leishmania actually resides in the phagolysosomes, where the pH is highly acidic. And so how does it survive in that acidic pH? So these mastigotes are somehow adapted to survive and thrive in acidic pH. So even when you're doing a test tube kind of experiment, like if you're growing axenic cultures, promastigotes will grow at neutral pH, but amastigotes will only grow at acidic pH. So again, the lysosomal compartment has a lot of acid proteases, a lot of enzymes, I beg your pardon, a lot of enzymes which could attack the parasite. So Leishmania actually secretes out certain cysteine proteases which will work in that acidic pH and which will destroy those enzymes, thus allowing it to survive. Other than that, so how does the macrophage mount the response? Again, by producing reactive oxygen species, right, which will attack these parasites and kill them. So Leishmania also has a way to try and abrogate the production of these ROS, the reactive oxygen species. One way it does this is by preventing the NADPH oxidase complex assembly. Okay, so there are actually many different ways. I think this is true for all of these pathogens which manage to survive in the macrophage. They have a multi-pronged approach. So these are just some of them which I've told you about. See, how does it infect macrophages? Again, there are so many receptors through which it can infect macrophages, but one of them is actually the complement receptor, CR1 and CR3. So it is able to actually use the immune system to gain entry into the macrophage and then survive.
Thank you, madam. I think there are many more questions. For want of time, we will reserve all these questions for a later point of time.
Yeah.
And once again, I thank you, madam, on behalf of all of us here for such a detailed presentation and well-informed responses on Leishmaniasis. Thank you, madam. Sai Ram.
My pleasure, and I'll be happy to join you all again. Sai Ram. Sai Ram.
Sai Ram. Now, we will have a very quick break and assemble back at 3:25 for the next session to start. Sai Ram. Om Sri Sai Ram. With the divine blessings of our most beloved Bhagwan, I welcome everyone for the fourth session of the first day's proceedings of the International Symposium on Sustainable Health. Now, I request Dr. Anand Kumar Sir from the Brindavan campus of the department to introduce the next speaker. Over to Brindavan. Sai Ram.
Om Shri Sai Ram. Offering myself with love and gratitude at Bhagwan's lotus feet. It's my honor and privilege to introduce Dr. Ravishankar Ramachandran, Head, Molecular Structural Biology Division, Chief Scientist and Professor, Central Drug Research Institute, CDRI, Lucknow. An alumnus of the Sri Sathya Sai Institute of Higher Learning, he passed out of the portals of the institute with bachelor's and master's degree in physics. In 1993, he joined Professor M. Vijayan's lab at Molecular Biophysics Unit, MBU. In 1999, he was awarded with the Alexander van Humboldt Fellowship at Max Planck Institute, Germany, where he worked in Nobel laureate Professor Robert Huber's lab. He then joined as a senior scientist at CDRI in 2002 and steadily grew in his stature, and he is currently the head Molecular Structural Biology Division, CDRI. He has received several honors such as National Bioscience Award for Career Development, Young Scientist Award from National Academy of Sciences, Best Publication Award from We Life Sciences. He is also a member of several academic and regulatory bodies such as Academic Planning and Developmental Committee, APDC of Naipur, Hajipur. He is also a member of Indian Crystallographic Association, Joint Secretary and Executive Member. He is also a member of Working Group on New TB Drugs, WGND, and so on. He has more than 80 publications in reputed journals like PNAS, Nature, Nucleic Acid Research, JBC, JMB, Cellular and Molecular Life Sciences, etc. He has about 9 patents and more in the pipeline. Dr. Ravishankar is the principal investigator for conducting the DCGI approved phase 3 clinical trials. Phase 3 randomized, double-blind, placebo-controlled trial of efficacy, safety and tolerability of antiviral drug Umifinovir versus standard care of therapy in non-severe COVID-19 patients. Dr. Ravishankar has developed several kits which aid scientists working on DNA metabolism and TB therapeutics discovery. He has guided several PhD students, about 15, and 7 more are presently working under his guidance. With this brief introduction, may we request Dr. Ravishankar Ramachandran to address us. Sign off.
Ram, it's a great pleasure and thank you for that warm introduction. Am I clear? Hello?
Yes, sir.
Yeah, thank you. Because I'm speaking from Manipur today and the internet is kind of flashy. I'll share my presentation right now.
Yes, sir. Please, sir.
Yeah. Okay. I'm going to speak to you today about a very interesting enzyme called phosphoserine phosphatase that is involved in serine metabolism in mycobacteria. My laboratory is a structural biology laboratory. I use a variety of structural biology techniques to answer interesting questions in biology, primarily using the TB bacteria as my favorite beating child. Yeah?
Can we have the presentation in full screen mode, sir? Presentation mode.
I have actually shared it in full screen mode. I've actually done it. So
It's okay, sir. You can proceed, sir. It's fine, sir.
But I have come to the second slide. Is it at least showing the second slide?
No, sir. No, sir. So probably you may have to exit from the Google Meet and then share the entire screen.
Okay, I'll share the entire screen. Yes, I'll do that.
Yes. You'll have to exit.
Yeah, sure. I'll just do that.
Yes, sir. Present now. Then the entire screen.
Mm.
In present.
You see the screen now?
Please click on present now on the screen.
No?
Sairam, sir, can you please click on
I did
present?
that.
Yeah, now it is coming. Wait.
I did that.
Yes, sir. Yes, sir. Now click on your PPT, sir.
Okay. I did that. I think it just some time. And on a slide it says tuberculosis is a deadly infectious disease. Are we there?
Not yet, sir. Not yet.
So what is the first slide?
No, sir. We are not able to see your slides. Yes. Yes. I think click on hide, sir.
Okay.
Click on hide down. Can you click on hide?
Okay, I do.
Down.
And I think more. Did that. Now is it fine? You can see tuberculosis is a deadly infectious disease.
Yes, sir. Yes, sir. You
Okay,
can.
wonderful. Yeah. So, like I told you, my lab is working on structural biology aspects. So we use a variety of techniques like X-ray crystallography, small angle X-ray scattering, cryo-electron microscopy, high resolution, and allied techniques to answer biological questions. Now, MTB is one of the most successful pathogens known to humans, primarily because of its ability to hide and evade the immune system. The etiological agent remains in a dormant state in our host macrophages for many years. And when our immune system goes down or other factors come into play, it gets into a full-blown disease. And as you can rightly imagine, regulation and adaptation during this kind of stress conditions it faces inside the human host is very, very important. So the enzyme, the hero of today's talk, is SIRB. RB number is 3042C. It is part of the essential L-serine biosynthetic pathway, and it is in the last step where it converts O-phospho-L-serine to L-serine and completely pathway. But what's so exciting about this particular gene in MTB is that compared to classical phosphoserine phosphatases, as in this slide I've shown you the domain organization, you can see that the classical phosphatase domain is shown in green, and in MTB you have two so-called ACT domains at the end terminus. And these adopt a ferredoxin fold with a beta alpha beta beta fold. Beta refers to the beta sheet and helix and so on and so forth. And there are two such domains in tandem at the end terminus. In MTB, there are two phosphoserine phosphatases, SERB1 and SERB2. SERB1 does not have these two ACT domains and has only the PSP domain. And knockouts have shown that it is not essential. SERB2 and for back. we started the characterization of these two genes and narrowed it down to MT SERB2 because it is more interesting for us, and because it's an unusual family phosphatase. So we tested it on a variety of substrates, L-phosphoserine, phosphothreonine, etc. I will run through some of these characterizations to get to the more meatier parts of my lecture, as I have only 45 minutes. It catalyzes the reaction O-phospho-L-serine or LD or D-serine, and results in L or D-serine plus the release of phosphate. And this is a close-up of the active site of which is there in the PSP domain. And you can see how these substrates kind of fit into the active site. And the active site itself consists of several important residues, the serine 273 and ASP 187, 185, 345, these several aspartates and aspartic acids. And if you take a series of peptides, it also utilizes phosphorylated peptides as substrate. And serine phosphopeptide, if it is considered to be of 100% relative activity, we have activity against threonine phosphopeptides and tyrosine phosphopeptides also at 25 and 5% relative activity. Then the next thing, if you want to understand all about somebody, we want to understand about your friend, you poke him here and there in the waist or something and make him giggle, or either he will smile at you or he'll shout at you. Same way with enzymes. So we fiddle around with some residues in the active site, mutate them to identify which are the important ones for activity. So this is what we did. We went through a series of mutations and some important motifs. And if you look at HAD family phosphatases, there are three classical motifs, which are called motif 1, 2, and 3 respectively. And these consist of important residues. So each of these were mutated and their effects were seen on the activity. If in this graph, which I plotted, relative activity versus the different mutants, if SERB wild type is 100% active, you find S273A almost negligible, double mutant D185, 187 in negligible, whereas the single mutants have partial active at around 10 to 20%, and so on. Let's look at the sequence analysis of the ACT domains itself, which I mentioned before occur in tandem at the N-terminus. The first ACT one is highlighted in blue box. The following one is in green box, and the three important motifs of the active site are also marked in this schematic sequence analysis. And as you can see, there is a lot of sequence variation in the ACT1 and ACT2 domains, whereas the phosphoserine phosphatase core domain, the active site domain, itself is very highly conserved with many conserved residues. The PSP domain itself is capable of hydrolyzing and utilizing L-phosphoserine as a substrate, but this is with much reduced efficacy, 6-fold or 3 to 6-fold reduced efficacy compared to the full-length enzyme. So the full-length enzyme, the ACT domains play a role in the activity. We figured it as allosteric, and the domain itself alone, we had these domain mutants, PSP domain alone. It is also active near neutral pH and physiological temperature, similar to the full-length enzyme. Whereas the ACT domain is the amino acid binding regulatory domain. So we carried out isothermal calorimetry experiments and showed binding of serine, and we saw that it binds quite well to the ACT domain, and you have this phosphoserine phosphatase domain binding also out here. And we solved the crystal structures of the enzyme from Mycobacterium avium, M. avium. The M. tuberculosis enzyme was not amenable to crystallization. We had very tiny crystals. We had better diffracting crystals with the M. avium enzyme. And you can see the electron density mapped as a mesh, and a cartoon representation of the close-up of the two active sites of the two serine binding sites of ACT1 and ACT2 domains. Now it so happens that these two domains can also bind to a variety of amino acids, and what we did, as you can see here, this experiment is with the individually purified domains of this enzyme. We have the SERB2 itself, the full-length enzyme. We have a relative activity of the PSP domain alone. And as you can see, if the full-length enzyme is 100-fold, this is about 40. And if you just add physically purified ACT2 to this PSP domain, it kind of doubles and reaches about 70% of the activity of the full-length enzyme. Then we added a variety of amino acids to probe their effects on the phosphatase activity of this enzyme. Some of them activate the activity. Native SERB2, if it is 100-fold, we have lysine, which takes it to 140. We have tyrosine, which again is nearly around 140. Serine itself acts like an inhibitor. And tryptophan, glutamic acid, and the remaining you can see in the graph. We did a series of elegant experiments where we showed using native gel as well as size exclusion chromatography and dynamic light scattering that when the active enzyme, which is a dimer, binds to serine, it becomes a tetramer and becomes inactive. So you can see it on the left hand side. The SERB dimer, suppose it is 100% active. Here the SERB2 tetramer, which is almost inactive. And these were pulled out from the size exclusion chromatography experiments, and immediately these were performed. So this initial part of the lecture, where the characterization of empty SERB2 was carried out, we identified it's an unusual phosphatase. It can also utilize small phosphorylated peptides. Of course, the one with serine was the most active one. And there is a specific transition which we identified to higher order oligomers. And this occurs upon the addition of L-serine at 0.8 molar ratio. This is the transition point between a dimer to a tetramer, inactive tetramer, which we demonstrated by the isothermal calorimetry and the size experiments. And the mutational analysis of these important residues showed that the transition is dependent on binding of L-serine to the ACT domain. So the binding of L-serine to ACT domains at the N terminus, not the active site, triggers this oligomeric transition. Furthermore, the higher order oligomeric form is inactive. So it's in the biological system, it is a mechanism for feedback control of enzyme activity. So there's an elegant way where there's a transition of a dimer to a tetramer, which is inactive. And again, if the concentration of this falls on, it will come back to a dimer and start its activity again. We did crystallize both the PSP domain alone, the full-length enzyme also, and compared it to the E. coli asensine domain alone. And these domains by themselves, the ACT domain, which is similar to what is called a RAM domain, a regulator of amino acid metabolism, it occurs in several transcription regulation proteins, allosteric regulators. Also, it occurs as part of standalone regulatory domain. So these are very interesting small domains. We solve the structure of the empty SERB2 ACT domain crystal, and this was to a resolution of about 3 angstroms. And I'll just run through some of these slides to show the structure solution. Then we also solved the crystal structures of the full-length SERB2 and its variants, that is its mutants, co-crystal structures with serine, and so on and so forth. And the crystals were frozen and data were collected both on synchrotron and the home source. And these resolution, the resolution of this data sets range from about 1.88 to about 1.9, 2.1. They're quite good. And the pattern was analyzed, and what we found was there is an extensive domain swapping in the oligomer. That is, the two ACT domains. If you see follow the one in the violet and dark blue, you can see that one of the ACT domains is swapped to the other side, and it kind of forms a very strong intimate bond, not a single bond, association. And this is the way it associates. And this is a close-up of the active site of the enzyme itself. We have several important residues there which were mutated, and this shows the differences that in the active site conformation. Going ahead, this is to show you that the data were carried out. We also got a hint from a report of homologous protein in gingivalis, and that's a pathogen which causes dental infection, Porphyromonas gingivalis, and that led us to look at possible new functions for this enzyme for interacting with human phosphoproteins. So what we did, we took the whole cell lysate, cytoplasmic fraction was isolated, and the cell envelope fraction was isolated. We also have had the antibodies against this enzymes. As a control, we took XTHA, which is an enzyme which occurs in base excision repair, which is not supposed to interact with random human proteins and not exported out. And then when we looked at it, we found that this protein localizes with several proteins and the cytoplasmic fraction. We also have a model system where we have uninfected THP1 human cells and also MTB infected THP1 cells. So here again, the bottom left, you can see that in uninfected THP1 cells, you cannot find MT-SERB2 in the cytoplasmic fraction. You find it only in the MTB infected THP1 cells, clearly showing you that this is kind of exported into the human cytoplasmic fraction where it is inside. Beta-actin was used as a control. And similarly, we had other experiments with CFP10, which is a virulence factor in MTB, in addition to actin. We also did these experiments with bronchoalveolar lavage samples from patients, and demonstrated it again that this protein is now exported. So these are totally new functions which were not known for phosphoserine phosphatase. Like I told you initially, it is predominantly working only in L-serine metabolism. And these are confocal images where you can see that in the presence of MT-SERB2, if you add this to such THP1 cells, it changes the cytoskeleton architecture. Whereas the inactive enzyme, I showed you initially a double mutant which was almost completely inactive, D341N, D345N. That is quite similar to the control or the non-added thing, showing that this whatever it is due to the activity of MT-SERB2 and it needs phosphatase activity. And so in this infection model, we looked at possible interacting factors. One is it alters tubulin or it co-localizes with tubulin, and as you can see with this overlay, it occurs in MTB as well as in the cytoplasmic fraction, as you can see here. So MT-SERB2 we found interacts with components of the cytoskeleton, and it mediates dephosphorylation of host cofilin. As you can see in this cycle, it can lead to actin remodeling through a variety like covalent phosphorylation. Would lead to this interaction with G-actin, and then R23 with the involvement of RAC with GTP, followed by actin polymerization. And these are supported by all the pull-down assays and the other assays for interaction with P-covalent and covalent also, showing that very clearly it interacts with the cytoskeleton. Then also we did a set of experiments where we looked at its interaction with several host phosphoproteins. We also looked at its interaction with HSP90, chaperone, 70, 27 GAPDH, and several interleukins, and we found that it interacts with several human proteins to elicit these different effects, primarily modulation of the cytoskeleton. Being in a drug discovery institute and having done a lot of characterization of this important essential enzyme, we were looking at, we carried out a virtual screening campaign against known and approved drugs, FDA approved drugs. And in this campaign, one of the compounds that we predicted was clofazimine. And clofazimine is known to be was used earlier as an anti-leprosy drug. And more recently, it has started garnering attention even by the Global TB Alliance for activity against, in clinical trials against drug-resistant TB, TB, clofazimine. Well, what is known is that it kind of interacts with and downplays ROS and that part. Mechanism was truly known. What we have done is, we believe, identified one of the important targets of clofazimine, and in this series of experiments, what we have shown is the binding to clofazimine through ITC using a series of both mutant enzymes and a native enzyme, and as well as a series of confocal experiments. We showed that the effects of cytoskeleton remodeling are abrogated by addition of clofazimine, which would obviously inhibit phosphatase activity of MT-SERB2. And this is similar to the effect we saw earlier with the double mutant, right? And clofazimine alone doesn't elicit this one. And clofazimine is a potent inhibitor of MT-SERB2. We showed that unambiguously here. And we then identified several proteins. This is more recent, not yet published here. And what we did is we did the human proteins and started looking at its interactions with the protein structural biology as well as context. And these are the down assays as well as assays with antibodies. And here in this history pull down analysis, we found that it forms a wonderful complex with the N-terminal domain of the P65. We used plasma resonance assays to characterize the binding kinetics of with the N-terminal domain protein of P65. We showed it in 18 moles, and P65 came so the internals and a small angle extension of the carried out to show that we have and well-folded protein. And as you can see that the Sachs envelope guided model is a good fit and fits well with the homology model of P65. And the next set of experiments were with MAP kinase P38. And again here with the purified recombinant proteins MT-SERB2 and MAPK P38, we showed that they interact very well with the Kd code 12.6 micromolar. Again here we carried out surface small angle X-ray scattering experiments with MAP kinase, and it's a good fit with the model, small angle X-ray scattering derived model. So a summary of the new functions of MT-SERB2 with the host include that it's essential for mycobacterial survival, and while this is known, and also that's an important component of the L-serine biosynthetic pathway, what the present work, the new work we carried out, showed that it is secreted out into the external milieu, and in one of the functions, dephosphorylates cofulin, and then this leads to the modulation of genes involved in cytoskeleton dynamics, and ultimately leads to cytoskeleton remodeling. This kind of then would facilitate invasion, so it can act like a kind of a invasion. Then in the other set of function, it dephosphorylates P38 and NFKB65 and leads to downregulation of IL-8, etc. And this immune system suppression. It interacts with important chaperones like HSP90, 70, 27, and GAPDH. Some of these have been also been shown to be important for the membrane interactions and modulation. And very importantly, we showed that MT-SERB2 is a target of clofazimine. Based on this, we signed an agreement with the Global TB Alliance for evaluation of several immunophenosyns, which are close analogs of clofazimine. And this was a important enzyme as an anti-TB target. And we are focused on now solving the structures of those multi-protein complexes of several host proteins like NFK, P65, and MAP kinase to see what exactly they modulate. And hopefully, in the longer term, we will have inhibitors which can then now target the host pathogen interactions based on this target and take us to a new therapeutic strategy. Thank you. Hello.
Thank you, sir. We'll now open the house for the questions. First, any questions from the Vrindavan campus?
I saw that at many of the
Yes, please go ahead.
Thank you. No questions.
Please go ahead, brother. Vrindavan. You're not audible. Mic should be on, I think. Just check the mic.
Hello.
Now you're audible. Go ahead, brother.
Yes, sir. Saram, sir. Sir,
Yeah.
what were the which were used to create the domain of the
Can you repeat?
There's actually an echo.
Could you repeat your question slowly, brother?
Saram?
There was an echo.
Saram, sir. Sir, what were the techniques which you used to crystallize the domain of the enzyme, sir? Protein?
We use a variety of techniques, but this was technique crystallized by what is called a hanging drop method. Crystallization of protein is a kind of an art. So you try several upon some condition which gives you small crystals, and then you start optimizing it. This was crystallized by so-called hanging drop method.
Thank you, sir. Any more questions from the Vrindavan campus?
Sairam, sir.
Yes, please go ahead.
why was the THP1 domain used for? Why was it? Sorry, why was the THP1 cell used for Mycobacterium tuberculosis?
It is used to mimic the macrophage. These are the cells which mimic macrophage. Model THP1 alone and THP1 cells.
Saram sir, because of some network issues, I
Yeah.
think there is little low transmission. Could you please switch off your video and then use only audio so that there will be little lesser bandwidth used? Saram sir? Internet. Saram sir?
Yeah, Saram.
Yeah, I think there is some network issue. Could you please switch off your video and then use only audio such that the bandwidth.
Yeah. I switched off my video.
Yes, yes, sir. Then now you can, yeah. Presenting also you can turn off, sir. We can do that. Okay.
See, in a village in Manipur. So
now it's better, sir. Now it's better, sir. Yes, sir. We understand. Sam, brothers, we will wait for a minute for Ravishankar sir to join.
Used in Mycobacterium tuberculosis.
Yeah, see, we wanted to mimic the macrophage. So we use the model, the THP1 cells, as you know, are the macrophage cells. And we use the model where we have THP1 cells alone and as well as the infected THP1 cells with MTB. So we are using that as a model system.
Okay, thank you, sir. Any questions from the Anantapur campus? Sarah, madam?
No questions.
Okay, madam. Thank you. Sir, one last question, sir. So
Yeah,
towards the end, you said that so the future directions would be to understand the entire protein complex and
Yeah.
then so try to solve the structure of it.
Yeah.
So what technique are you thinking of, sir? So such a huge
High
complex.
resolution, yeah, high resolution cryo-EM.
Exactly. Okay. Thank you, sir. That was also I was thinking the same line. Thank you, sir. So cryo facility, where do you think we can have such a facility like in India?
Well,
Like
there is one very close to Puttaparthi in Bangalore at the INSTEM. It's a
NCBS.
42 to 40. It's a very high resolution facility. That's about 42 crores. It's a national facility that's close to you. There's
Yes.
a 200 kV facility at IIC Bangalore, which is again nationally, it's a
Yes.
national facility. These two are close to you. There's one at Hyderabad at CCMP, so on and so forth.
Right, sir. Thank you, sir. Probably we will explore using them in the near future.
Sure.
Yes, sir. Thank you, sir. Thank you very much, sir, for your wonderful talk on structural biology. So, in fact, last time when we had met Professor Vijayan, me and my student, when we had gone to IIC to present, so Professor Vijayan was such an enthusiastic person. Like he always told that, so we are in need of a lot of crystals, and we have synchrotron facility in Indore, and all of you are welcome to come and solve the structures of a lot of proteins. Youngsters, like he was such a infectious. His enthusiasm was so infectious too. Just be with him itself is a joy, sir. So thank you, sir. Thank you very much for your wonderful presentation. And
Okay, sir.
sir. Sairam, moving ahead, I request Dr. A.S. Viswanathan, sir, Assistant Professor, Prashant Nilayam campus, to come forward and introduce the next speaker. Sairam.
Sairam, good evening, everyone. It's my pleasure to introduce the next speaker for today, Dr. Govind Rao, who's Professor of Chemical and Biochemical Engineering at the University of Maryland, Baltimore. Professor Rao completed his BTech in Chemical Engineering from IIT Madras and went on to complete his PhD from Drexel University at Philadelphia in the year 1987. Ever since, he's been a faculty member at the University of Maryland, Baltimore, and he has served as the department chair from the years 2000 to 2006. He founded the Center for Advanced Sensor Technology in the year 2006 and has been serving as its director since inception. Dr. Rao's research is targeted towards disruptive innovation with the primary objective of creating paradigm shifts in the state of art. His research group has been making giant strides in sensor technology, focusing on applications to reduce costs associated with healthcare and to close disparity gaps by making innovative, low-cost devices for use in low-resource settings. His group has also made valuable contributions in the area of point-of-care biomanufacturing. To tell you a little more about Professor Rao, he is very diverse in his research applications. He's obtained research funding from many different sources, which include the National Science Foundation, National Institutes of Health, the Juvenile Diabetes Research Foundation, the Defense Advanced Research Projects Agency, US FDA, Bill and Melinda Gates Foundation, and many other companies. He's published over 180 papers in professional journals and has several patents, many of which have been licensed. Dr. Rao has served on various government and industry advisory panels and has also given several invited keynote and plenary lectures at various international conferences and corporations. He's been the recipient of several awards, which include the Presidential Young Investigator Award from the National Science Foundation, Outstanding Teaching and Research Awards from UMBC, and he's been named a 2003 Innovator of the Year by the Maryland Daily Record. Coming to more recent times, in 2015, he received a 50 Bright Ideas Award from UMBC for the number of inventions filed, and he was named an eminent engineer by Tau Beta Pi. His innovative cardboard incubator won the 2019 Academic Pediatric Association Global Health Research Award. And he has recently been named the 2019 Presidential Research Professor at UMBC. He has also served as the chair of the Biotechnology Division of the American Chemical Society and on the editorial board of several journals. To top all of these achievements, He attributes all of these to the blessings of our most beloved Bhagwan. And to conclude on a personal note, having interacted with him very closely as his doctoral research student, I have personally learned many life lessons from him. I wouldn't want to stand between you and Professor Rao anymore. Professor Rao will be addressing us today, and he'll be speaking to us on the topic, Disruptive Innovation for Sustainable Health. Over to Professor Rao. Thank you.
I'm now in presentation mode. Are you able to hear me?
Yes, so we are able to hear you, sir. And not in the full screen.
I'm sorry?
Presentation is on, not in the full screen, sir. It's not in the presentation mode.
Okay, no, I will put it, I'll switch off my camera for bandwidth and just go. I just because when I'm in the presentation mode, I can't see anything else. So are you able to see it?
Yeah.
Yes?
Maybe once you go to the presentation mode,
I am in the presentation mode.
we don't see that yet.
You don't see it yet. Okay. Are you able to see the first slide?
Not yet. I think it's coming now.
Okay. Are you able to see it?
No, not the full screen mode yet. We're still able to see the slideshow mode is not yet.
No idea. How about now?
Not yet.
Okay, so let's just go in this mode. You're able to see the slides, right?
Yes.
Okay, so we'll just do it in this mode. So, my pranams at Swami's lotus feet. This is obviously all through his grace that everything has happened. And much of whatever I talk about is all inspired by him. And in terms of how we focus on research, I can't say it better than Swami can. And this is a quote you probably are all familiar with. And Swami has always exhorted us to make sure that our work is of benefit to somebody at the end of the day. And there's no point in just doing research for the sake of research. So this has been our touchstone and our guiding principle. And we've really tried to make a difference by using something called disruptive innovation. And it's much easier to immediately grasp what a disruptive innovation is if you simply look at how the landscape has changed over the past 20 years. Are you able to see my slides as I'm advancing?
Yes, sir.
Okay, so all these are why we're able to even connect today and sort of, I'm able to make this presentation from the other side of the world. And there have been truly transformational technologies. But again, all of these took a great deal of imagination to come up with a concept that simply didn't exist. And I've wondered if the same can be applied to healthcare and to truly come up with advances that make it affordable and accessible to anyone. This is where I am. I'm at the UMBC campus, which is the University of Maryland, Baltimore County. The University of Maryland, Baltimore, where the medical school is not far. It's just about 10 miles away. And this is the building that I reside in, and all our labs are. I started a Center for Advanced Sensor Technology because I realized that research to be truly impactful needs many disciplines to tackle complex problems of today. And so this was set up to be a hub of disruptive innovations with a focus on reducing healthcare costs. And what makes it possible is our ability to integrate biology, chemistry, physics, electronics, and molecular biology all under one roof. And very focused by a quote that Dr. Martin Luther King has said, very much along the lines of what Swami has taught us. Life's most persistent and urgent question is, what are you doing for others? So over the years, we've been very fortunate to receive many visitors. This is an example of Norman Augustine, who's one of the most distinguished engineers. And he actually is somebody who has a very astute eye on the broader impact and how cost can enter the equation. And one of the things that we've focused on is just how unsustainable the healthcare cost is. Now, this is an old slide, pre-COVID. And if you add the impact that COVID has had globally, it is absolutely dragging us to a point where you cannot afford healthcare. And the other motivator has always been to think of what Gandhiji has said, recall the face of the poorest and weakest man, and really focus ourselves on what can we do to help improve the lives of people. Here's another grim statistic in the US, and that is that people's income now has reached a point where you are spending more than you're earning. And so you're basically reconciled to a life of debt. So all these factors have conspired to create almost a catastrophic situation. If you're somebody who has a healthcare plan or insurance, you're okay, but the average person who has to pay out of pocket is in dire straits. So this is where disruptive innovation comes in. And again, disruptive innovation is simply a way of thinking about something in a way where you have a trajectory of change that's transformational. So normally technology progresses in increments. And so, for example, telephones were landlines, then they switched to digital exchanges, but really nothing changed much in terms of the number of people who had access to telephones until mobile phones came. So that's an example of a disruptive innovation where you rapidly expand the pool of users. So this is what we're trying to focus on for healthcare. And this is something that is just beginning. So you now currently have to go for advanced scanning to the super specialty hospital or its equivalent anywhere. But as the technology is moving, it is moving downstream in terms of the skill level and the places that you can apply it. So rather than a major hospital, eventually you can do it in your home. And so even the skill set needed. So for example, Professor Sanjeevi kicked off this symposium with his talk on diabetes. Central to diabetes is glucose monitoring. And now you can monitor glucose with a small glucometer in your home by sticking your own finger. This was not possible 50 years ago. You had to go to a hospital. So this is the kind of innovation and change that's needed. With regard to biopharmaceuticals, this is a typical footprint of a biopharmaceutical plant. You have bioreactors where you cultivate cells, recombinant cells in the tens of thousands of liters, and they grow for weeks to days, and they're secreting a heterologous protein, which then must be recovered. So the cells are typically lysed if it's an intracellular product like an inclusion body in E. coli, or if it's excreted like by Chinese hamster ovary cells, you still have to recover the product, purify it, stabilize it, and ship it. And this is what is happening worldwide. A good example is the Serum Institute and its amazing capability to manufacture billions of doses of vaccines. But the problem is you need very expensive, large infrastructure to achieve this. And so I was fortunate that my paths crossed by Swami's grace with Dr. Jeff Ling, who is a retired US Army officer. He's a doctor. And one of his earlier claims for innovation is he's the one who created this concept of a thought-controlled prosthetic arm. And this has now been approved by the FDA. So the idea was for amputees to regain their functionality and be able to have a normal life. So when Dr. Ling was in Iraq and Afghanistan, he was very frustrated that he was not able to obtain medicines. So he also happened to be a program manager at DARPA. DARPA stands for the Defense Advanced Research Program Agency. It is the arm of the Department of Defense that funds crazy ideas. And they're very, very proud of the fact that they take risks. They're very proud of the fact that 85% of their ideas are failures, but the 15% that succeed are game changers. So a good example is our ability. My talking to you today is because of DARPA, because they're the ones who created the internet. Your ability to find your way anywhere on the planet depends on the GPS system. And that is, again, something that was a DARPA innovation, because nobody had imagined that such things would be possible. So he wanted to do the same to healthcare. And he said, look, I'm tired of this large footprint facility that's manufacturing drugs and vaccines at some remote site, and I'm unable to get it where I need it. So how can we come up with a system that produces medicine from a very compact footprint setup? So he challenged us to think of ways where you could make it compact and something you could produce it at the point of care. So right by the patient's bedside or in a village, anywhere in the world, you should be able to make these medicines. So again, it was fortuitous that we came across this whole field of cell-free expression. And cell-free expression is fascinating because it turns out that if you take any living cell and strip it of its nucleus or coding DNA, all the organelles and the machinery that's left behind, it turns out that it's still functional and can take basically any foreign DNA and transcribe it and translate it into protein. So why would you do this? The reason to do it is basically when you're doing recombinant protein production, there's fundamentally a conflict. The cell evolutionarily is programmed just to divide and grow. It doesn't really want to produce your heterologous protein. You're forcing it to by putting in a gene. And as far as the cell's concerned, evolutionarily, it just wants to survive and not really produce your protein. So there's an inherent conflict where you wanted to produce the heterologous protein at the maximum possible yield. So if you can break that conflict with a cell-free system, you basically use the machinery, and then you no longer are focused on all the thousands of genes that are necessary to keep a cell alive, and you can focus on just a single transcript. And so this makes it very, very rapid. The other remarkable thing is that you're also able to make a lyophilized product. You can actually bust open the cells and make these extracts so that they're freeze dried. And just by reconstituting with a buffer, you're able to get the protein expression happening. And this is just showing you quite dramatically how a lyophilized pellet, if you just add buffer and then add GFP, the plasmid, it starts expressing. And what's even more remarkable, this is not a typo, the process is extremely fast. So in 2 hours, you're getting your maximum expression. That's because you're no longer constrained with the cell having to grow and divide. You're just using the transcription and translational machinery of the cell. And it's so amazing. So very often we have visitors in the lab. These are Department of Defense people who just are active duty military personnel, and we show them that anyone can do this. These two gentlemen are actually pilots. In their day job, they're flying F-16s, and they wanted to learn more about the programs that are funded by the Department of Defense. And in 2 hours, they were making protein. So our first prototype basically was this suitcase-sized device. And again, this is where interdisciplinary approaches are very important, because not only do you need the molecular biologist who can do the expression, but you also need an electrical engineer who can put the hardware programs and controls. You need mechanical engineers who can build the platform. You need computer scientists who can program and automate the script. You need in our center we have people with all different backgrounds. It's truly a team effort. You cannot work in isolation anymore. So what this suitcase does is it has everything built in, and it takes care of the complete purification in a completely automated fashion. And as you can imagine, there's a script that you can run, because the other thing that you can do is automate this. So all of these steps, which normally you would do painstakingly with spin columns and washes and recoveries, all those are automated, and the computer takes over and does everything. And we showed how we can make it very reproducibly. These are data from 2 different suitcases producing GCSF. And you can see that all the fractions collected give you essentially the same result. So you've automated the manufacturing of a protein. Now, next generation, everyone's talking about Industry 4.0, you build in even more robustness. And these are sort of next generation approaches using machine learning, artificial intelligence, etc., to make sure that your product is very consistent and very pure. This is just John Scully, the former CEO of Apple and Pepsi. So he had visited the lab to take a look, and he was taking a photo on his iPhone. So I sort of was happy to see the capturing of 2 disruptive technologies by a former CEO of Apple Computer. And it was very gratifying to hear him say that this is one of the most innovative things he's seen. So while we were doing this, Zika happened. And so Zika is now no longer in the news. It's all COVID-19. But at the time, Zika was posing a huge threat. And what we were able to show is that you're able to essentially clone and express a piece of the Zika envelope protein in under 24 hours. So the idea being that you could do very rapid vaccine development. And our vision was, if this technology were available in a distributed fashion throughout the world, if there is an outbreak, you can nip it in the bud by quickly producing vaccines or neutralizing antibodies or whatever, and at least treat it right where it's happening. So imagine if this were available in Wuhan, perhaps the infection would have been nipped in the bud and we wouldn't be facing this catastrophic situation right now. These are just examples showing how the microfabrication works. We're able to make things like Humira, which is a monoclonal antibody. It's the largest selling antibody. It's used for all kinds of inflammation-linked diseases. It basically inhibits tumor necrosis factor, TNF-alpha, and prevents inflammation. So it's one of the most widely used for rheumatoid arthritis and psoriasis and many other inflammation and immune disorders. So this is how we imagined, in collaboration with Johns Hopkins, we had a focus group. How would medicines of the future be manufactured? And perhaps they might be made in cassettes, like printer cartridges, where you just push it into the machine and it will produce different proteins. All the ingredients are built in to these cassettes, and everything is done pretty much like your smartphone has different apps. You would have different apps that would run the script, and it would all be to ensure that the right patient gets from the right prescription, all barcoded, so that you don't give the wrong drug to the patient. And this is all data that has been published, if anyone's interested. And as you can imagine, it's also attracted a fair amount of press, where the suitcase has been featured as a futuristic concept. We wanted to push it further and actually show that we could make FDA-approved drugs. So we chose a radiation medical countermeasure, and that's called Neupogen or filgrastim. This is basically granulocyte colony factor, GCSF, macrophage factor, and it basically helps you recover your blood count after your bone marrow has been destroyed due to a radiation incident. So it's used for people who are receiving chemotherapy, for example, or radiation therapy for cancer, but also if there's a radiological accident, such as a Fukushima disaster, or if there's a terrorist-related dirty bomb, you would need vast quantities of this to save people's lives, to help them deal with radiation sickness. So we just showed how it was possible on a daily basis to manufacture this protein and every day come up with enough doses to treat mice. We did a mouse study. Are you still seeing my slides?
Seeing, sir.
Yes, sir. Okay. So
Yes.
this is a study that we conducted as a proof of principle. So there were 40 mice taken. 10 were not treated, and then 30 of the mice were irradiated to simulate a lethal dose of basically 70, lethal dose of 30, meaning that 30% of the animals would die. So once that was done, we had a control group that received just saline, and then one that received the GCSF on a daily basis. And you can see how the study progressed very rapidly. In about a month, we showed that the mice that were treated with the GCSF had 100% survival, whereas the ones with saline showed about a 20% death rate. So biological variation, we had planned it for a 30% death rate, but 20% died. And so this proved the efficacy of this approach. And this is, again, all published data that's available. But the point I want to make is this is the science, but there's also an application that one must keep in mind, and that is, how is it impacting a human at the end of the day? You know, Swami is always telling us about things that eventually affect and help others. And this was happening while the study was going on. One of the people in the lab had cancer and had to receive the same GCSF that we were making. This is a prescription for Neulasta, which is a different version of the Neupogen or GCSF. And they had to take 5 injections, each one of which was $12,000. So over $60,000. They had insurance, so they didn't have to pay anything. But imagine some poor person who doesn't. How would they afford this? And so that is the target. We're trying to make sure that we can make it so it doesn't have to be so out of reach. So let me switch gears now to another topic. And this has to do with neonatal applications and how I got into this whole cardboard incubator that Vishwanath talked about. So we had been working in collaboration with General Electric Healthcare to come up with a non-contact temperature sensor. So the idea being that you could simply put a gel and measure a baby's temperature remotely without having a wire on the baby, because when the baby moves, the wire detaches, the signal is lost, there's all kinds of problems. So this led to a lot of interest, and the CEO of GE came to the lab, and Jeff Immelt at the time, he said, this is really interesting. I'd like to fund you to come up with low-cost sensors that will allow you to do non-contact sensing for other analytes, not just temperature. And so the idea was, can we do sensors that will measure various physiological parameters without any the trauma? And the reason for that is very simple. If you look at this, this is the current way in which you get samples of blood from a baby. So you have to stick the baby's heel with a lance. That doesn't sound terrible, but if you imagine the scale, it would be like you or me, adult-sized humans, being stabbed in the heel with a 2-inch knife. Now suddenly it takes a different perspective because that would be obviously extremely painful, and you can see the face of the baby reflecting that. So, and if you want to get meaningful data, you have to continuously make these measurements, which means you have to keep stabbing the poor baby's heel. So we then had this idea that maybe it's possible to look at glucose that's diffusing across the baby's skin, and using our sensing technologies, maybe we can detect it non-invasively. So the idea was to place a buffer against the skin and then collect the glucose that diffuses across the skin over a period of time, and then measure it. We have a very sensitive sensor based on the glucose binding protein from E. coli that's fluorescently labeled. And so we're able to see conformational changes in the protein when it binds glucose. And so this is a great example of how protein engineering and chemistry and optics all come together. So to give you a solution to a major problem. And this was actually tested and validated with adults, where the transdermal glucose shown in blue was validated against the red points, which show actual finger stick measurements of the glucose. And so this was also done with both a normal and a type 2 diabetic to show that the correlations were pretty good. So this is technology that can now be used with other analytes that could be diffusing across the skins. And maybe there's plenty of other biomarkers that we can go after. Another approach has to do with how you can measure CO2 and oxygen, and it's the same principle. So here it's a two-stage approach where basically you circulate nitrogen. Let me just move this out. Yeah, okay. So you have a two-stage approach. You flush the sampler that's held against the skin with nitrogen. What that does is give you a zero baseline. So you get this baseline here. And then you flip a valve and you recirculate the gas past an oxygen and CO2 sensor. So what's happening is the sampler is held against the skin, and now oxygen and CO2 that's diffusing across the skin basically are picked up by the sensor. And by simply measuring this slope, the slope which gives you the rate of diffusion across the skin, mathematically applying Fick's law has to be proportional to the actual arterial blood concentration of CO2 or oxygen. So this rate-based measurement gives you a simple way to measure arterial blood concentration without actually needing a blood draw. You need an initial blood draw for the calibration, but that's it. After that, you can just do it. And here, these are data. So this is what the sampler looks like. And it's just a small hollow chamber that you hold against the skin. And this actually, let me just show you. This is a 24-week baby weighing 520 grams. So the physician is holding that little sampler against the baby's leg. This is the entire baby's leg. Okay, look at it. It's smaller than the finger of the doctor. The entire leg from thigh to foot. So that's how tiny these babies are. And the normal way you do it is you stick a needle. And any neonatologist I talk to tells me that that's the worst aspect of their job, trying to find a vein in a structure this small. So we did the comparison, and you can see that this transdermal measurement of oxygen and CO2 correlated very well with the actual blood measurement. So hopefully this will reduce pain and suffering for lots of infants and find widespread use wherever Swami decides to take it. Hopefully this will be of use to society. Now, while we were doing this, we also work a lot in cell culture, and rather than having to do all the experiment on babies, we thought, okay, why don't we use our bioreactors and put the sensors outside of a diffusible membrane, and that way it's easier to refine our instrumentation. And so we've built many different things. So people who are doing cell culture, like Pradeep, for your chondrocytes and other things you grow. Now, in tissue culture flasks with a simple dialysis loop, you should be able to measure glucose, glutamine, and then oxygen, CO2 in all these vessels. And that's exactly what we've done by having a simple little loop that you insert into these tea flasks and spinner flasks and whatever apparatus you might use for cell culture fermentation, or as Vishwanath did for wastewater. You can then easily know what's going on. You can look at the oxygen and CO2 with a great deal of accuracy, completely non-invasively. So this will give you insights into the metabolic processes that are going on in your culture system or the human body or in a wastewater treatment facility, just to make sure that the system is performing as it should. Again, to switch gears, Swami is always talking about ensuring that the helpless are helped. And so our approach to Seva is really to see how we can expand healthcare access. And so here is sort of the kind of snapshot of the world population that lives on less than $10 a day, which is 71% of the world population. And clearly, there are people who are, even below $1 a day. So how do we make sure that all these amazing advances in science and technology impact the common man? And here's another depressing statistic. Every 10 seconds, a baby, a newborn, is dying somewhere in the world. That is tragic, but it becomes even worse when you realize that it's a preventable death. And most of them are due to things like hypoxia, sepsis, and asphyxia, etc. So this is obviously a huge problem, and we thought, can we do something to tackle it? And again, I was very fortunate to have a team that we went and looked at different sites in rural healthcare centers. And this is the former VC, Dr. Shashidhar Prasad, who helped do this. This is Dr. Hanumantappa. Again, these are philanthropists and physicians who have made it sort of their life mission to improve the lives. And this is me with a bunch of students and faculty colleagues. And Mrs. Geeta Mohan Ram, who many of you may know, she served as the translator for this team. And we interviewed the physician, and this is what they use to keep a baby warm. It's a table lamp. So this is at a small healthcare facility outside of Mysore. And so what the students did was they interviewed the frontline nurses and workers with Mrs. Ram as the translator into Kannada. And they basically told us that, we had an inexpensive cardboard incubator that we had designed, the students had designed, and wanted their feedback over what is the feature set. See, this is the other very valuable thing. Most people who offer sort of help, they don't consult the people who actually are the frontline workers, the nurses, the midwives. And so this was eye-opening because they gave us all kinds of input into design features, which we eventually incorporated. And again, we were grateful to Mr. Shashi Kumar, again, a connection made by Swami. Mr. Shashi Kumar is the son of Mr. Unni, who is in charge of the accommodations in the ashram. And he is the managing director of Phoenix Medical Systems, India's largest manufacturer of incubators. And we were very gracious, and they agreed right away to partner with us and serve as the manufacturer for this cardboard incubator. And this is the actual incubator. It's a design that folds and flat packs that you can unpack and assemble in the field, and it's now ready to receive a baby. So it is remarkable, and by Swami's grace, we did a non-inferiority trial with 48 babies in a control arm and 48 babies in this cardboard incubator, and showed that our incubator worked just as well in terms of temperature regulation. But even more important, the hospital incubator, which had to be cleaned on a regular basis, had 2 instances of drug-resistant bacteria, but the cardboard incubator had none. So imagining its use in the millions, you could see statistically how the prevention of sepsis might be something that these achieve, because they're single use. Once you're done with one baby, you discard it. And we have also taken great care to make sure that all the components are biodegradable so that you don't cause a landfill problem. So this is where this stands. So the idea was, can you take this one step further and build sensors to monitor various parameters of the baby? So in this case, the image and the weight. So the idea being, you could then conduct telemedicine applications. And so the idea would be, you could have a system where these incubators could remotely send data on the baby. For example, you can tackle malnutrition by using machine learning, artificial intelligence algorithms. If the baby's weight gain isn't happening on track, then an ASHA worker or someone can go and check up on the baby and make sure. And I was lucky to then meet with Nandan Nilankani, show him this concept. And he basically, their primary focus is the banking, but certainly we've got now the connection to follow and do some healthcare applications as this moves forward. So again, a lot of this work is done by students, undergraduate students who work in teams. They're working on the prototypes of the incubator, etc. I didn't have this in my abstract, but with COVID-19, I just wanted to share a very simple way that we've come up with to assess what the threat might be from SARS-CoV-2 at any time. And this is, all of us know this now, that masking is probably the simplest way to prevent transmission. That's because it's aerosolized droplets that contain the virus. So we thought maybe there's a simple way, if we can just collect the aerosol in the atmosphere by condensation, just use a dehumidifier and then measure what's going on, we might be able to get a signature for what the actual threat level is. So right now in your auditorium, if you're all sitting there, and if one of you is unknowingly shedding a virus, you wouldn't really know till the others got infected. But if you had a system that would use a simple dehumidifier that is condensing the air and couple it with a rapid test, you would actually immediately know that, hey, there's SARS-CoV-2 in the atmosphere, let's vacate this place or let's ventilate it or whatever. So we just used a little something that's readily available from Amazon for about 2,500 rupees. And we deployed it in the hospital at different locations. Okay? And they were deployed for 1 week periods and sampled every day. And the idea was that you take this sample and extract RNA and do an RT-LAMP assay or RT-PCR and a protein-based ELISA. So unfortunately, we didn't find any RNA because just the way it degrades very quickly, and we're only taking it from the condensate once a day. But we did find ELISA gave us positive hits, and the number of positive hits increased even more if we used viral transport medium in the condensate capture tank. So the idea now, eventually we imagine something like this, where you have a wearable that monitors the atmosphere around you and tells you how many infectious particles there are. So hopefully this technology will save even more lives and help us deal with this pandemic situation. So with that, I'd like to come to a close and really thank all the taxpayers of India who gave me almost free education, and the taxpayers of the US that have basically been funding a lot of this research. And really a large number of students, postdocs, somewhere here, Vishwanath's name is here, and also Sai Satish, Venki, then Professor Ravishankar Sai. So you can see that it's really not just me, it's Swami who has provided all this ecosystem around us to make sure that this work happens. And so with that, I'd like to conclude, and I'm happy to answer any questions.
Sairam sir, thank you very much for such a smart presentation, I would say. And now we are open for questions. Firstly, if there are any questions from the Anantapur campus, I request them to pose. Madam, ma'am. Any questions from the Anantapur campus? Moving on to Brindavan campus. Any questions from the Brindavan campus?
Sairam sir, no questions from Brindavan.
Please go ahead.
Sairam sir, no questions from Brindavan.
Okay. Sairam sir. So here is a question that is posted on the chat box. So what is the scope for nanotechnology in advancing easy portability, accuracy, and economy of manufacturing units and medical technologies?
I think it has tremendous potential, but not directly, more in the field of diagnostics, because these nanotechnologies are very useful, for example, quantum dots and other reporter systems that allow you to visualize your analytes and make measurements in a much more sensitive fashion. So it's very much part of our toolbox to keep pushing the technology applications forward. How do we incorporate ELISA in a wearable device? With microfluidics. So a lot of our systems are designed with microfluidics. If you Google the term wood microfluidics, you'll see something that Venki worked on, where we just built microfluidic systems out of wood to serve as a low-cost approach to do a lot of flow chemistry and analytics on.
So one question from here, sir. Sir, you just mentioned that so in order to analyze the glucose level in the blood, so non-invasively, so you said that you put a strip and then you have the, if you have a buffer, so in the container, and then there is a transfer of ions across the skin. So is it any different from the iontophoresis that we generally come across, and then so to pick up ions and then even try to measure ions in that?
Yes. So the approach we're using is completely passive, Naresh. So you don't need, with iontophoresis, you have to actually put electrodes and supply a current to draw out the ions. In this particular case, it's passive diffusion. So the measurement becomes even simpler. Literally, you're washing the skin. That's all you're doing.
So in which case, do you think there will be scope for non-invasive glucose sensors like wearables on the skin and then as a patch sensor? So
yes,
that taken somewhere?
Yes, that would be the concept. So we're also looking at different targets. So for example, if there's a biomarker for cancer, for example, you could potentially have a patch on the skin that you monitor every now and then to make sure that there's nothing happening in the body. It would not completely replace, but at least minimize the blood draws.
Yes, sir.
Naresh, we have a question from Anantapur.
Sir, yes, please go ahead, madam. Please go ahead. Are you able to hear, sir? There is a question
Yes.
from Anantapur campus. Yes, ma'am.
Sir. First, we're extremely grateful that we got to hear this kind of a presentation, because we are really gaining some new perspectives about how what we study is actually used in the real world. And as an undergraduate student who is really inexperienced in the field of research and application, my question is extremely basic, sir. If we are passionate about science and we want to go about it in the way that you are, sir, innovating so beautifully, how do we train our minds to think in such an interdisciplinary fashion and combine knowledge from more than one walk of science?
Very good question. I'm so glad you asked it. Too often we over specialize and we don't observe the world around us. So if I had to give you a one-word answer, curiosity. Be curious. Always strive to understand how something works, why something is done a certain way, and don't be afraid. Be curious, be fearless, and never be afraid of asking a question. I'm so glad you said very basic question. I tell all my students, there is no such thing as a stupid question. The only stupid thing is if you don't ask the question. So we're all learning here, whether it be in the path of spirituality. Even Swami emphasizes Atma Vichara. Ultimately, that is what you need to do. You need to be asking the why, the what, the how, the who, and that will lead you. So also that's one tip. And on a more practical note, don't just read the literature in your field. The most of my advances and ideas have come when I see what's happening in another field, and then I see that, my God, they have a solution that will solve my problem in this field, and that immediately lends itself to applications that you can do. Good example, many basic scientists have studied these binding proteins. Wonderful crystallographic studies, structural studies that go deeper and deeper. And it was those fundamental studies that we said, wait a second, the fact that people have seen in the crystal structure how this glucose binding protein changes its conformation when it binds its substrate means we can make a sensor with it. And so we just put a fluorescent label on the protein that basically changes its fluorescent signature depending on the hydrophobicity of the environment. And lo and behold, when the protein is binding glucose, the atmosphere around that fluorophore is changing from hydrophilic to hydrophobic, or vice versa, depending on where you tag it. And immediately, as it binds glucose, we see this wonderful shift in the fluorescent spectrum. Now you then make an optical reader for it, and you've suddenly created a super sensitive glucose sensor. And now there's a vast array of binding proteins that we're targeting with a similar approach. So just one example. Then the other is even more basic. If babies are dying due to hypothermia, basically all you need to do is keep them warm. And yes, you can wrap them in a blanket and everything, but there are many places where night temperatures can be quite cold, and really babies need an incubator to kind of stay warm. And the cost was prohibitive when we found out what happens in most of these rural centers. A, they can't afford it, or B, some well-meaning NGO will donate an incubator, but after about a year, when it breaks down, it's useless. It's just a piece of hardware that's gathering dust that no one can use. So understanding what's appropriate, what's needed, and using your scientific knowledge, tailoring it to that is what it's all about. I hope that answers your question.
Yes, sir. Thank you so much, sir. I think you've inspired a lot of us to change the way we think about science and how we want to use it to help other people. Thank you so much, sir.
No, I'm so happy to hear you say that. I keep telling people, just spend 5 minutes a day. Just think about, what it is that you've done and how is it going to impact somebody else. I mean, this is the heart of what Swami's teachings are. Just that sort of will lead you to observe things in a different way and come up with solutions that potentially could help make life better for someone.
So I have a question. Sir. You have done lot of work in this area. So do you have prepared any sensor related to the nerves being so sensitive and intricate? The sensors were prepared for the nerve systems also, sir? Nervous system.
So what our sensors, we have made measurements of neurons and astrocytes growing in culture and made measurements of the oxygen levels around them. So certainly to optimize and understand what the conditions are to grow these cells in culture, our sensors work.
Thank you, sir. Thank you, Naresh.
We have one more question here from Prashant Nilayam campus.
I have one very small add-on question to the previous question that you answered. You were telling about the importance of going beyond the discipline that we belong to, but given the conventional setup that we students study in generally, like biology, physics, chemistry, and mathematics, how would you suggest that we make a beginning to start exploring other disciplines which we don't generally have access to till we are students?
Organize yourselves. Unfortunately, the academic structure, we tend to build walls around us. So you as young faculty, research scholars, a good example is this symposium. It's sponsored by the Biosciences Department. Physics had its own, and chemistry will have its own. Consider joint symposia, consider a journal club where people present ideas of something that they've read. Don't have it just for the department, have it for everyone. Open it up exactly like the symposium, extend the invitation to everyone. I'm a good example of this even in Parthi, right? So you were my PhD student, you're in the bioscience department. On your advisor, your co-advisor was Professor Ravishankar Sai, who's a physicist. You used extensively electrodes and electrochemistry from the chemistry department. So literally, it's like the three gunas, physics, chemistry, biology, and mathematics. All these have to be part and parcel of what you do, and just build on that. So create this ecosystem where you have general talks where people talk about ideas and you understand what's going on in each other's disciplines. If you're forced to explain the fundamentals of what you do to a non-specialist, it also allows you to hone your communication skills. How do you explain something that's very specialized, very esoteric, very complex in a way that's understandable? That's Swami's life, his message, exotic Vedanta that he made so easy to digest for all of us. Same approach can be brought to bear here. So hopefully you'll be leading off sort of a forum where you all present results and discuss them, seek feedback from each other, and be open. The one question, whenever I give a seminar, I am much more interested in the criticism than in sort of, okay, you gave a wonderful talk, because I want the basic scientist to tell me that, hey, you didn't think about this, or the chemist to say, what about this? You never considered it. And that allows me to then stretch and reach out to the next possible problem to solve.
Thank you very much, sir. And if there aren't any more questions, so I can only say that that was an icing on the cake, and your insights have been so invigorating, sitting somewhere thousands of miles across. So you're able to inspire us, and then I'm sure many of the students who have connected, so you have started them thinking and how to integrate multiple subjects, and then how do we think they can be on the path of disruptive innovation, being the students of basic sciences. Thank you once again, sir, from all of us. And we have been observing, like, from the morning, you were there, and then I'm sure, I don't know whether you have slept or not. Thank you very much, and it's already early morning, and maybe again you are back to your work. So with this, we come to the end of the proceedings of the first day's proceedings, that is our International Symposium on Sustainable Health. And tomorrow, so we will be assembling again at 11:20 a.m. To continue with the second day's proceedings of the International Symposium on Sustainable Health. So now, we will close the day's proceedings with prayer. Shanti, Shanti, Shanti. जय बोलो भगवान श्री सत्य साई बाबा जी की जय.